Everyone, and welcome to today's webinar on Northwest Agriculture Agronomy Update. If you have any questions today during the presentation, you can type them into the question section on the GoToWebinar control panel, and we will answer them at the end of the presentation. This webinar is being recorded, and you will receive a link to the recording. Thank you.
Good afternoon, everyone, and thanks for tuning in virtually today. We sure hope to get to have these meetings in person again soon, but for now, we're going to take advantage of the technology to share information. I'm Nicole Clausen, and I'm the Farm Production Extension Specialist for Crops in the Swan River office, and I'm co-hosting today's webinar with Boris Mikuleski out of Dauphin. I have thrown up my number on the slide. If anyone has any issues or questions, feel free to text me anytime. Boris, do you want to say hello quickly? Hey, maybe we're not still muted or something.
Good afternoon, everyone. Thanks for joining. And I know that it's a little bit different from usual forums that people have been able to attend, but we hope you get the information you need virtually.
All right, so below I have our order of speakers, and I threw up approximate times because we are here for the afternoon, and in case there's a certain specialist that you want to be sure not to miss, just for you to refer back to. So the idea of today's webinar was agronomy questions that were around the Northwest region. Even though we haven't seen many producers this winter, we wanted to just let you know that we're still here and we're ready to answer your questions. And we posed several questions to our specialists, so they're going to go through in their presentations. In the interest of time, let's get started with our first speaker. Ann Kirk is our provincial feed grain specialist, so if she wants to take it away, Lori, I'll get you to turn the screen over to Ann, please.
Okay, I think my screen is being shared now. Thank you for having me today. So there's a couple of questions that I'll be answering. The first is a question that came in was, why are some Canadian Western Red Spring wheat varieties being moved to the CNHR class, so the Canadian Northern Hard Red class? And is there a risk of older varieties of other crops being deregistered? So to answer this question, yes, there are some varieties that are moving to CNHR as of August 1st, 2021. So that means that while these varieties are still classified as CWRS right now, by the time harvest comes, if someone has these 5 varieties in the ground, they would be classified as CNHR once they're harvested. So this was indicated in the seed guide, and this is the second time that that's happened in the last couple of years. So as of August 1st, 2018, there were 25 CWRS varieties and 4 Canadian Prairie Red Spring varieties that were moved to this class. So the reason why these varieties are being moved is because of the Canadian Grain Commission Wheat Class Modernization Program. So that program happened because gluten strength was declining. So customers of CWRS and CPSR wheat were concerned about their gluten strength, and it wasn't as high as required and a little inconsistent. And since it had been declining in recent years, the Canadian Grain Commission decided to address this by strengthening the quality parameters around CWRS and CPSR. So they revised the quality parameters to narrow them up a bit to ensure that the quality of those classes was being maintained. So as a result, the CNHR class was created in 2016, and varieties that didn't meet those more strict quality parameters were moved to the CNHR class. So you know, I think this had to be done to maintain the consistency and quality of the Canadian Western Red Spring class. And really the characteristics that they're looking at was just the milling, dough strength, protein, and end-use functionality. So while the varieties in CNHR will have slightly lower gluten strength than CWRS varieties, and they have a wider range of protein levels, and the CNHR varieties, will they still offer good milling characteristics? CWRS is maintained as the gold standard. So that's the reason for this. And as of now, there's no plans to move other varieties to the CNHR class. But you know, these stricter quality parameters are being considered at the time of variety registration. So in terms of other varieties of other crops being deregistered, there is a process for canceling varieties with the Variety Registration Office with CFIA. So if a registrant of a variety does want to cancel a variety registration for whatever reason, they have to send a request to the Variety Registration Office. And that requester has to ensure that seed stocks of the variety are removed by the end of the 3-year notification period. So, you know, people wouldn't be growing or selling any more certified seed. This is, you know, more of a concern to farmers that are consistently saving seed. And in order to try to alert people, CFIA has to post the cancellation notification list on their website and then send that notification of cancellation to the Canadian Seed Growers Association, the Grain Commission, CFIA. I did check the CFIA cancellation notification list recently, and if you're interested, you can, you know, just Google CFIA variety cancellation, and it will come up. In recent years, or this year, there's a wide range of soybean varieties that are on the list, and that's what I did notice the most of is some soybean varieties. There definitely are some cereals and other crops, but the ones that are on the variety cancellation list do tend to be ones that you know, wouldn't be commonly grown, like they're being removed, usually just because there isn't interest in growing them anymore. So that's the variety deregistration process. So the next question that I had was for spring wheat. So what row spacing and plant populations should be used to promote uniform growth and less tillers in wheat? So in general, more narrow row spacings and high plant populations will promote less tillers and therefore more uniform growth. So when there's less space, so when varieties are more evenly spaced, or sorry, when the plants are more evenly spaced and they're packed tighter together, there's less resources available per plant, and then therefore less chance of tillers. They're just, when there's more space, more light, more resources for a plant, for each individual plant, they will produce more tillers. When there's less space and resources, they will not produce as many tillers. So in Manitoba, we recommend a target plant stand ranging from 23 to 28 plants per square foot, and I would definitely recommend targeting the high end of that seeding rate, so around, you know, 28 plants per square foot. There are some farmers that are targeting higher plant populations, so anywhere in that 30 to 35 plants per square feet, and they might have specific reasons for doing that. So it is important that agronomically optimum seeding rate is something that's talked about quite a bit. And it is important to think about this for each individual farm. So below optimum seeding rates could reduce resource use efficiency and yield, whereas above optimum, you might be increasing your cost in production for no reason. You might not be getting any yield or other benefits, but just putting more seeds in the ground. And there could be also yield decreases due to diseases, insects, or lodging. So in Manitoba, we did do seeding rate studies at the crop diversification centers. Well, in general, I think looking at this graph, you can see that there is a bit of a trend towards higher yields with higher target plant densities, but there weren't really statistically significant differences in yields across a range of target plant densities for wheat at the higher target plant densities. So ranging from say 21 to 39 plants per square foot, there wasn't a huge range in yield. So, and I wouldn't expect, so farmers also that are planting at those really high densities, I don't think they're expecting, you know, higher yields. They're likely doing it to reduce tillering and try to even up the plant stand. But there are some situations where farmers might want to increase yields or increase seeding rates. So what's interesting is that the agronomically optimum plant density can vary by both environment and variety. So by variety, each variety does have a differing ability to tiller. So some varieties do perform better at lower seeding rates, while some varieties would perform better at higher seeding rates. We don't really have a lot of that information available because it's, you know, pretty tough to do a lot of variety by plant population studies. And the studies that we've done in Manitoba, we haven't really seen any significant differences in variety, in yields by plant population between different varieties. But what I wanted to mention is that there has been some of this work looking at agronomically optimum plant density across a range of environments. And there was a study that was released last year that was, they did an analysis, this was from Kansas State University, and they did an analysis of existing literature. And they also concluded that plant density should be different at varying yield environments. So when the yield potential of an environment increases, the plant density could be reduced. And this kind of goes across against what some people may have thought or may have given some thought previously, is that, you know, in those high yielding environments, you should increase the amount of inputs to get a better return. But these studies are saying that in higher yield environments, that you could actually reduce the plant population. So in this study, just to give you an example of what they're thinking about, they did find that in high yield environments, the optimum plant density was 16 plants per square foot, whereas in low yielding environments, the optimum was 37 plants per square foot. So I would never recommend that anyone seed at 16 plants per square foot, but I just think it's interesting to consider how much that optimum plant density could differ across a range of varieties or across a range of environments. And there was one that's probably more relevant for us, where they did a large seeding rate study at North Dakota State University. That's the one highlighted in gray here. And they did find that across all environments, a seeding rate of 32 seeds per square foot would be appropriate, whereas they found the same thing. In low yielding environments, you should increase the seeding rate, and in high yielding environments, you could decrease the seeding rate by a little bit. And just to keep that in perspective with our target plant densities, 38 seeds per square foot would be a target density of 26 seeds per square foot. So they're planting 38 seeds per square foot, and with taking into germination and mortality into account, you'd be ending up with a target of about 26. So that'd be the higher end of our seeding rate. And then, oh, sorry, I did that wrong. Across all environments, the 32 seeds per square foot would be a target of 26. And in low yielding environments, planting 38 seeds per square foot would end up with about 32 seeds per square foot. So just something to consider when thinking about, you know, the time of seeding, environmental considerations, and that optimum plant density. So times when you might want to increase seeding rate is if you have, if you're looking for more weed competition. So if there's hard to control weeds, or organic farmers would be increasing their seeding rate to help compete against weeds. As I already mentioned, uniform growth with less tillering. So for fungicide staging and even maturity. And then also, just to tie in that lower yield potential, if you were, say, seeding at a late date due to environmental conditions, or you were reseeding wheat past the optimum time, I would definitely recommend increasing the seeding rate, because your yield potential would be lower, and therefore the wheat would be less resource efficient. And just in terms of calculating seeding rates, I did mention expected seedling survival. So it's important to consider expected seedling survival, and that will differ from location to location, year to year. And also the 1,000 kernel weight, I think it would differ, you know, the same variety might differ by 20% from year to year. So it is important to make sure that you know your 1,000 kernel weight for every seed lot that you're planting. So the row spacing effects was the second part of the question. And generally with cereal crops, narrow rows are generally recommended, but studies do show various effects of row spacing on wheat yields. So in some cases, studies have found that there is no effect on yield with varying row spacing. Recently, Rob Golden at the University of Manitoba, along with a grad student, have been doing row spacing studies in spring wheat. And they did find that those narrow row spacings did result in 15 and 20% greater yield compared with a 15-inch row. So 15-inch is pretty wide, but it's just, you know, good to know that that research is showing that those narrow row spacings do result in higher yields. One some studies by Guilafond in Saskatchewan and Manitoba, though, did show that when they were doing zero tillage, there was a slight benefit of having wider rows. And I think just having to do with the stubble and the way the crop was planted, and for some stubble management effects, they did see some slight yield increases with wider rows. But under conventional tillage, they didn't find any effect of row spacing. So the second part of the wheat question was how to manage wheat in terms of nitrogen for high yield and protein. So the most recent nitrogen recommendations that we have in Manitoba are from the research done at the University of Manitoba with Amy Mangan and John Flayton, where they looked at 2 different wheat varieties at a variety of locations. They applied nitrogen at varying levels, and with that study, they were able to identify an economic optimum nitrogen rate for fertilizer, the yield at that optimum fertilizer rate, and then the total nitrogen supply per bushel. And based on all of that research, they have identified that the optimum soil test nitrogen plus fertilizer nitrogen rate is 2¼ pounds of nitrogen per bushel. So that's down slightly. Previously, what was generally recommended was about 2½ pounds of nitrogen per bushel. And with newer varieties, they did have found that they would recommend 2¼ pounds of nitrogen per bushel. And I do think it's important just when you're setting these yield targets and determining nitrogen rates to try to make sure people are actually setting realistic targets, because there wouldn't be any benefit to setting a really high target, applying excess nitrogen and applying excess nitrogen. So, and the other part of this is both timing. So if you're looking at targeting both high yields and high protein, timing of nitrogen application can be important. So first of all, in order to maximize yield, nitrogen fertilizer does need to be available at the time of maximum crop uptake. So for cereal crops, that's the start of stem elongation, which does happen, fairly early in the growing season until heading. So in this research done at the University of Manitoba, they did look at applying nitrogen at planting, stem elongation, and a flag leaf application. So splitting those applications between both planting and stem elongation, or planting and flag leaf. The first bar graph with the blue bars does show yield results from those different timings of nitrogen application. And there was a slight increase in yield with that stem elongation split compared to planting, but the real difference does come with protein. So they did find with applying nitrogen later in the season, they were able to increase protein, but not necessarily see that yield increase. So those delayed applications do tend to increase protein without increasing yield. And that's because it's being applied at the time when nitrogen is being relocated into the seed. It's not necessarily being used for additional plant growth. But this is in part important when targeting high wheat yields, since there is that inverse relationship between yield and protein. So that's all for wheat. The final questions I had were seeding rates for oats and fertility to maximize oat yields. So for oat seeding rates, generally in Manitoba, we recommend about, say 20 to 25 plants per square foot of oats. It does really depend on, your conditions on your farm and what you're seeing. So wild oats do tend to be a concern with oats. Say about half of, many fields in Manitoba do have wild oat issues. So if wild oats are an issue, then I think increasing seeding rate is important. And also in a late seeding situation, it's also recommended to increase seeding rate a bit as well. So there has been quite a few studies looking at oat seeding rates to do with wild oat control. So this, the graph shown here is from a study that was conducted in both Saskatchewan and Manitoba at multiple locations. So they looked at both a low wild oat density, which is the solid line, and a high wild oat density, which is the dotted line. And in both cases, as seeding rate increased, the wild oat biomass, so the amount of wild oat plants in the field, did decrease. And in this research, for wild oats, they do emerge really early in the growing season. So a strategy that some people had used was, if you do have a lot of wild oats in your field, to delay seeding until wild oats can be controlled. So in this study, and we know with a lot of cereal crops that earlier seeding does result in the highest yield, especially with oats, where we do see concerns with quality in terms of the plumpness of the seed with late seeding. So we do recommend early seeding with oats. And if there is a situation where there is going to be wild oats in the field, this research did show that early seeding is possible as long as higher seeding rates were used. And they are recommending, that 350 seeds per meter squared seeding rate in order to get some good wild oat control. And you can see that there wasn't really a big difference in wild oat biomass between the 350 and 450 seeds per meter squared. This is just from the same study showing grain yield at those seeding rates. So grain yield did increase with increased seeding rate, and it was maximized at 350 seeds per meter squared. So this was from the study, and I think this makes a lot of sense that if there is a lot of wild oats in the field, then the seeding rate should be about 350 seeds per meter squared. And that does translate into a target of about 27 plants per square foot. But you know, if wild oat density was really high, then in some cases they were recommending higher seeding rates. But I think what's important is under conditions that were relatively free of wild oats, other research has found that seeding rate didn't have a significant effect on grain yield. So that was other research conducted in Manitoba and Saskatchewan, did find no yield effects with increasing seeding rates if there weren't wild oats in the field. And we did find similar results with the study that we did at the diversification centers in 2017 and 2018. The lines across the graph do show yield at a range of target plant densities at 5 different locations. And as you can see, those lines are pretty flat and do even decrease at some locations with those higher densities. And in this case, we didn't have any wild oat issues at any of those locations. There wasn't any weed issues. So we did see a flat response to increased seeding rate. So I think when trying to determine seeding rates for any of these crops, it's important to think about, what your target is, the reasons why you might want an increased seeding rate. And if you don't expect to have any issues with weeds, then sticking with a more conservative seeding rate like that, 20 to 25 plants per square foot as a target would be fine. And if there was wild oat concerns, I would suggest raising it a bit. The last question was on nitrogen fertility in oats. So the general recommendation, because of research that was done in Saskatchewan and Manitoba, did find that optimum yields were achieved with about 100 pounds of nitrogen per acre. And when I was, just ensuring that that was the general recommendation, I did look at a bunch of studies, some studies in Manitoba and Saskatchewan, and the range of recommendations was about 89 to about 110 pounds of nitrogen per acre. So going with 100, I think, is very reasonable. And some of these studies have found that exceeding 100 pounds of nitrogen per acre, so that would be both the fertilizer and the soil test nitrogen, didn't result in any additional yield. And higher N rates, with oats, there's a good chance that higher N rates will lead to increased lodging. There can also be reductions in test weight and fewer plum kernels. So I think there's going to be some studies starting this year looking at nitrogen rates in oats, just to see if some higher targets would make sense. But I think if people are looking at increasing nitrogen rates in oats, the general recommendation would be to choose really high yielding varieties with good lodging resistance. But I think that sticking with that 100 pounds of nitrogen per acre makes a lot of sense, or that is the recommendation. So, we'll see with this additional research if higher nitrogen levels, how they do. So that's all I have for cereal crops. And if anyone has any questions, I'll be happy to answer them at the end of this program.
Well, thank you for that, Anne. Our next speaker is Dennis Lang. He is the provincial pulse specialist. Now, anybody that's had the opportunity to see Dennis's presentation in person in the past knows that he usually wears his trademark Hawaiian style shirts. Unfortunately, we won't be able to see him model the all famous Hawaiian shirts today, but I am sure he will not. He will be able to give us all the information we need about all the pulses in Manitoba. So with that, take it away, Dennis.
Yeah, thanks, Boris. I'm going to give everybody a little treat here for a minute here, just for 1 second here, and see if it's going to work. There we go. One of the few times that you'll see me do a presentation without a Hawaiian shirt on. So, but we won't have it on for this one here. So, anyways, so we had a couple questions come across my table here with respect to the oops, hang on a second here. See what happens when you try to have fun. There we go. There we go. So we had a few questions on both field peas and on soybeans as well. So the first one we're going to start off with is on field peas, and we want to talk a little bit about fertility and disease management in field peas. One of the big things with field peas is good nodulation. I think that's really important when it comes to yield. It's very important when it comes to protein content. You need to know kind of what your history is, you know, of growing field peas. And the Swan River area, you have a fairly good history of soybean or growing peas in your region. And a good history means, you know, once every 3 to 4 years, or in some cases maybe once every 6 years, if you're dealing with possibly some phaeomyces issues. But for the most part, if you have some history there, then what ends up happening is you're not as reliant on the 2 different forms of inoculant. I always like to recommend to growers, first couple years that they're growing field peas, is you should have inoculant on seed and in furrow. So if you're using a liquid or peat on seed, you should also look at putting a granular in furrow, or in some cases growers have used liquid in furrow. It's a way of having 2 different, I guess 2 different forms of inoculant, so that if you have a wet year and the inoculant washes away from the seed, you still have that in furrow inoculant as a backup. So that's kind of why I like to recommend this having inoculant in 2 forms. Now, when you are using inoculant with peas and you're looking at doing a seed treatment, what are some of the issues you need to be aware of? Well, the inoculants don't have the same lifespan as what the soybean inoculants do. So it's really important that when you're inoculating, you inoculate as close to seeding as possible. If you are using one of these seed treatments, and on years like this, when the soil is a little bit on the cool side, I would recommend looking at a fungicide seed treatment. Going with that we'll call it double inoculation, so having it on in 2 different positions in the soil, one on seed and one away from the seed. Having those 2 forms there will allow more bacteria to function, I guess if you want to call it that, if you have a seed treatment. Sometimes the seed treatment can affect that bacteria, so having that in furrow inoculants is another reason why you might want to do that when it comes to seed treatments. The advantage of a seed treatment, you know, in most cases it's for things like Fusarium, Rhizoctonia, and in some cases early season of phaeomyces. If you have some insect problems, then you can also add an insecticide seed treatment as well. The complete list is listed on page 546 of the Guide to Crop Protection in the 21 guide. As far as fertility, other aspects with respect to fertility, you know, with phosphate, we talked about nitrogen, and one thing I'll mention as well, and I do get this question from time to time, is how much nitrogen can I have in my soil for the bacteria to really function properly? If you're under 50 pounds, you're fine. If you're 70 to 80 pounds, you're still probably going to be okay, but if you're over 100 pounds, then you might want to start looking at other crops. You want to take full advantage of that nitrogen that's in the soil, and peas in that situation, you won't get the full advantage of that. So under 50 pounds, perfect. 60 to 70, still okay, but you want to try to focus on fields that have low nitrogen for the most part. For phosphate, typically 30 to 40 pounds is a side band. You can't really put much down with the seed, only about 20 pounds with the seed. Potassium, if you are short on potassium, then you are looking at applying some on low testing fields in that 30 to 60 pound range. It is recommended doing a soil test to make sure you know what your levels are. In some areas like the Red River Valley, for example, potassium levels usually aren't too much of an issue. Whereas if you go into the eastern part of Manitoba and some of those peaty soils, well, then that's a little different story. So it's always good as a starting point. That's kind of what you need to look at is look at your soil test and look at where some of your deficiencies are. Just to add to that, I pulled this off the MPSG website, Manitoba Pulse and Soybean Growers. There's a recommendation of doing some starter phosphorus has shown to increase P yields even on high P soils. And again, that whole maximum 20 pounds of phosphate with the seed is about all you can really do. So, and it's kind of some of those things. Keep your full management fertility management package in check. Soil test to make sure that you're applying the correct amount. And for the most part, you should be very successful in that respect. One thing I wanted to kind of talk on here before we get into the next section that on field P is something that's kind of come up recently here. When you look at this pea plant, and this is something that I'm going to kind of go into a little bit here. These are the different stages of the field pea plant. Field peas have scale nodes. They're either at or just above the soil surface. And for any of the growers up in the Swan River area last year that had all that wind and really damaged the plants early on, that's where you start to get that regrowth. And that's why peas are so, they are all more frost tolerant, and they are more tolerant if you have damage early in the season. But what's important when you're trying to judge what stage the peas are at, let's say you're looking at spraying a herbicide on it and you have up until the 6th node stage. The one thing to keep in mind, ignore the 2 scale nodes. Start counting where you have these leaves here that are those clasping leaves. So in this case, this would be your first leaf. And in this case, they use a node, but the terminology is first leaf node, or just first leaf, and then second leaf, and so on and so on. If you're doing it that way, then you're fine. This scale leaf node, sometimes it will have a branch, and you don't count that in that case. So if it doesn't have a clasping leaf, don't count it. If it does have a clasping leaf, count it, and then you should be fine for your staging. So just something to keep in mind when you're doing staging. So harvest management in peas. So this is a slide from last summer. It was down in my neck of the woods here. The grower had not used any kind of harvest management product on it. And there was the odd green plant that was in the sample. They were harvesting about 17% moisture in this case. The sample looked very nice. Those odd green seeds that were in there, for the most part, dried down quite nicely in the bin. So it wasn't a necessity to spray the crop to help dry it down. This was in mid-August, and this is a 80 bushel crop of peas. Now you'll notice by these pictures, the ground's pretty flat down in the valley here. So we don't have those hills and valleys, and some years we can get away without desiccating. But in areas where you have more hills and valleys, desiccation becomes more of a question. So the first thing I want to talk about when you're talking about desiccants is I want to make the statement: glyphosate is not a desiccant. It's there for help to control weed products or weeds in the crop at harvest time. It helps dry those weeds down and kills the weeds, but it's not really a true desiccant. So there are a number of companies in Manitoba that are buying peas, Roquette and Merit Foods. You look at AGT Foods, you look at Best Cooking Pulses, and some of those companies as well. At this point, a number of the companies that are buying those products or buying peas for some of their markets, they've basically said right now glyphosate is not something they will allow on their crop. When it comes to other true desiccants, like a lot of the companies are either going with diquat or heat, and that's something you have options. Diquat is your Reglone, and there's various brands of that. Some of the things to kind of keep in mind between the distinction, if you're using that, like say diquat or heat. Diquat is something you want to apply when it's cooler on a cloudier day, and then you want it hot the next day because you want that product to get, it's a contact product and you want good coverage. So you don't want to be applying it on a really hot day. Heat is not as critical for those conditions. What is important for both is good water coverage, 15 to 20 gallons. Some of the sales reps that I've talked to, they're trying to push growers to that 20 gallons. In the past, growers have had the option of mixing with glyphosate. And unfortunately, for a lot of companies that buy peas right now, glyphosate's off the table. So growers have to look at other options. So if you have some issues where you have to look at using some of these products, best thing to do is check with your buyer. Diquat is something that the MRL has changed in the US as of last fall. So it has opened up. A year ago, diquat couldn't be used by almost most peat companies weren't allowing it. Now that's changed with that new MRL regulation, and they're going into some companies are going to the US where it's not as much of a problem. But you still want to check with your company that you're dealing with to make sure that the product that you choose to use on your field is going to be acceptable. Thank you. Okay, and we'll go to the next one here. Okay, so field pea diseases is another topic that has been brought up as well. And the Bacterial Blight is, of course, one of the most common diseases that you'll see in peas. That's what most growers tend to spray for. It can cause leaf and stem and foot rot. The pathogen is seedborne to airborne. It's spread by rain splash, and it can survive on the stubble for many years. There we go. Again, you know, if you have a nice dense canopy in that early flowering stage, and you have the right conditions, which we'll go through in a moment here, you're going to have to budget for spraying for a Microspirilla. And when you get to that point of spraying, you'll have some tools that you can use to help determine whether or not it's going to be worth spraying at that time, or whether you can wait a little bit. So here's a nice crop. This is the same field that you saw earlier in the harvest management photograph. This picture was taken in early August. Beautiful looking stand. And this is my summer student from last year. This field again did over 80 bushels. The interesting part of this, and I didn't have that photograph in this slide, but in this particular image here, the field that he had 2 miles away looked absolutely horrible compared to this. There, that field was a much, you know, a lot poorer drained, and that field looked like it was ready to harvest already. So again, field selection really important in that respect. But getting back into disease here, for fungicide timing, the ideal fungicide timing is at that early flowering stage. So roughly at about 10% flowering. But there's a nice checklist that you as a producer and industry can use to kind of give you a benchmark on whether or not it's time to spray now, or whether or not it's time to spray later on. We're just going to go right into the next slide here. And with this slide here, this can be found here at the website link listed here, but we're going to kind of walk through it here. So when you're looking at your crop canopy, you have your different risk factor scores here. So let's say our first score that we're going to use, we go out and we look at what our canopy is like at that 10% flowering stage, and we see it's very dense. So in this case, we're going to give it, well, we're going to give it a normal in this case. We're going to give it 8 in this case, instead of giving it the 30, but we'll say it's normal. So 8, it says lentils, but it's actually, this one is interchangeable with lentils and peas. So that's 15. We look at leaf wetness at noon, and there we see, give it a score of 20, which is moderate. So it's still pretty moist at that time. The 5-day forecast is calling for light showers. So that puts us at 15. Then we start looking for symptoms on the plants. And this is probably one of the things that we really need to look for is look into that lower canopy, look for those purple freckles is probably the best way to describe it, and see if you see those purple freckles in there. So in this field that we did here, we're saying up to 20% of the plant showing, and that gives us a score of 15. If we total that up, we come up with 65. That means in this case, you would be spraying. Now, if you went out and saw no visible symptoms, but you saw everything else in favor of disease development, you would probably want to wait, maybe come back, you know, 3 or 4 days later and see if you see any disease development, and then make that decision to spray at that time. The reason I suggest this is because on years when it's a little on the drier side, delaying that application is going to be beneficial for you, because then you may only need to spray once. Every field at some point will have Bacterial Blight in it, but the longer you can push it to harvest, the better it is, because once it's full pod set, then it's really not going to affect the yield at all. So that's maybe the purpose of this. In some cases, it might be pretty easy. You know, you're looking at moisture and you're looking at disease, and in this case here, it's pretty obvious that you'd want to spray. But in some years, and we've had it in 17 and 18 and part of 19 as well, where some growers have only had to spray once because they waited. And in some cases where the stand was a little bit thinner, growers looked at the numbers and said, you know, for the cost of it and the risk is low, I'm not going to spray this year. So it's just something to give you an aid in doing that for you to increase your profitability at the end of the day. The next section I want to talk about is soybean diseases, and the question was, does fungicide spray? So I'm going to talk a little bit about the diseases that are, it can be found in soybeans, and then I'm going to, at the end, show you a couple of slides on what the research has been done and what the results are. So the diseases we're mostly concerned with, Toria brown spot. There's a fungicide option for that. Bacterial blight. It's a bacteria, so fungicide application really won't help in this case. But it is good to know what you're looking at, and we'll talk about that. Faraga leaf spot, and also white mold are the ones I'm going to focus on today. Okay, starting off with bacterial blight. Now, bacterial blight is very distinctive when you look at the plants. A lot of times when I go out looking and I start looking in its upper to or mid to upper canopy, what I start to see is these brown lesions with a nice yellow halo around it here. And here's some different stages. And as it progresses, you can see here these lesions coalesce, and eventually they start to drop off. A lot of times with bacterial blight, I can pretty much, count backwards and determine about 10 to 14 days previously, we've either had some hail or some strong winds or some heavy rains. And that allows the plant, or that beats up the plant slightly, allows the disease to move in. But it's a bacteria, so fungicide really isn't going to do anything. A lot of times, the best course of action here is just to kind of leave it alone, and it'll grow out of it. And back probably about 5 years ago, there were certain varieties that I did see that were worse for bacterial blight, but those varieties aren't grown anymore. And now I think you can go out into almost any field and find bacterial blight at low levels. So again, something to watch out for, but not something of any economic concern at this point. Okay. So Toria brown spot, that is something you can do something about if your levels are high enough, and if you feel that it's a risk to losing yield. They're a little different. A lot of times they're found in the lower canopy. You don't have that halo around the brown spot. Eventually, it does take over the entire leaf and yellows off the entire leaf. But in most cases, when you look at compared to a bacterial blight, you can see here with the on the lower surface of the leaf here, just these brown specks, and on the lower surface and on the upper surface. But with a bacterial blight, you always have that halo around it. So again, just to be able to differentiate between the two. Okay. White mold. White mold, the last time we really saw any kind of white mold issues in soybeans was back in 2013. This is an image from southern Manitoba, and in this case here, canopy was really dense. We had cooler conditions right around that early flowering time, and here we saw quite a number of plants. You can see all these dead plants that have been affected by white mold or sclerotinia. The northwest up around that Dauphin area did see this as well. Digging into some of the research, so from down in North Dakota, and I'll show a slide on this in a moment here. I've read some reports. You need to be at least 10% white mold in your field to see any significant yield loss. So if you think about that for a second, 10% would be almost 14,000 plants per acre, so which is pretty significant. And with the shorter maturing varieties that we grow in Manitoba compared to what they grow in North Dakota, the risk for white mold is a lot less in the shorter season varieties than they are in the longer season varieties that they would grow further south. Okay. This is a slide from Michael Warsh from NDSU, just talking about what factors favor the particular white mold or sclerotinia development. Cooler temperatures, 15 to 25 degrees, and high humidity. Frequent and light rainfall will make things worse as well. Wet conditions during the bloom and early pod stage, typically in that R2, R3 stage, and rainfall leading up to the R2 stage can also facilitate apothecia production as well. One of the comments that he had made based on some of the work that he's done, and if any of you are interested, I can send you some results of those, but his comment too, when we started talking about white mold in Manitoba, was that the risk of disease will likely need to be very, very high for any fungicide application to be profitable. On average, the shorter season varieties grown in Manitoba are typically exhibit some moderate susceptibility to sclerotinia, but for the most part, we just don't see a lot of white mold in Manitoba. Faraga leaf spot is relatively new. We have found Faraga leaf spot in the various areas that we have that we've looked at. But again, it's never been at a level where it's been of any economic concern. You can see here that these lesions here, the circular triangular lesions, resemble the eyes of a frog, giving the disease its name. So what about the results? What have people been doing? So Kristen McMillan from the U of M, part of the Soybean and Pulse Agronomy Lab, they've done 3 years. They've used 2 fungicides, single and sequential applications. They've seen no significant yield responses. And again, those are 3 consecutive dry years, so that's something to keep in mind. As part of the on-farm network for Manitoba pulse and soybean growers, they've had 7 sites in 2020 with 3 different fungicide options, no significant yield losses. And their response rate since 2014 has only been 15%. 15% of the time is where they've seen any kind of yield benefit to putting a fungicide on, and that's done over multiple years, including 2016. So the answer to your question is, as far as whether fungicides pay, the research in Manitoba right now has indicated that it's not worth the going to spray with it. The one concern I have is a grower sometimes will tell me, well, I want to, crop looks nice and green, I want to keep it green for as long as I can. Well, remember that these soybeans are long season. Keeping something green longer than it should is not necessarily a good thing in Manitoba. We did have some frost in early September last year, so if there's disease present and significant there, you might want to consider a fungicide application. But so far, the results that have been done in Manitoba is showing no real significant benefit in doing that. So with that, I'm gonna close it off here. Put my contact information up on here, and also I'll put Nicole's up here as well. She's part of our Pulse team, and any questions you have on peas and soybeans, you can direct to her during the growing season. So I thank you for now.
Thank you, Dennis. That was lots of good information for our area, especially as we move into more acres of field peas. I also appreciate the bomber shirt as much as the Hawaiian shirt, and I really appreciate that it wasn't a Rough Rider shirt. Just a reminder, if anybody has questions or comments, you can type them into the box on the side panel, and we can get to them at the end of the day. And we do have, I forgot to mention that there is CCA, a CCA code at the end of the presentation that we'll get to you for anybody collecting credits. Our next presentation is from Dane, and he's our oilseed specialist. And I'll let Dane get into it because I hear he's got lots to share.
Hi, Nicole. Yeah, thanks everyone. It's going to be a bit loaded. I adjusted the speaking list from what Nicole had given me a little bit earlier. I wasn't going to address fertility so much, but I'm going to go into some of those hidden yield robbers. I know there was a couple issues in the northwestern part of the province this past year with canola that wasn't performing quite the way it was, and I wanted to get into examining that and seeing why that might be the case. Nicole, do you have questions that are coming up throughout, or would you rather have us address them all at the end?
I don't have any questions that have popped up yet.
Okay, the slides are looking good. You're seeing the main screen?
From what I can see, I think so, yep.
Super. Okay, then we'll just jump right into it. All right, so finding hidden yield robbers. It's never a clear-cut solution that I could tell someone to say, look at this and go look at that. This is kind of a diagnostic tool I use in my mind, and it's something I wrote down the last week or two, just giving a nice picture of how that thinking process works, at least for me. So it might work for you as agronomists and farmers as well. So when I'm looking for a solution, or there's something wrong in the field, one of the first things I start looking for are sources of human error. We are our own biggest downfall in many aspects. So I'm looking for things like straight lines, overlap patterns, geometric shapes, things that are a dead giveaway that it wasn't the natural occurrence. Once I can rule things like that out, was it the right implement, miscalibration, gaps, doubles in seed bed establishment, those sorts of things. If that can be avoided, I'm going to start looking at the soil and the seed bed. Was it too wet or too dry for the implement at hand? Too wet, was seed trench smearing, compaction, were there too many residues on the surface? Was it too dry? Was there a lot of soil moving around, a lot of soil drift, piling up in shelter belts and ditches, things like that? Moving on from there, Then I'll start looking at environment, moisture patterns. What has it been like for the past 2 weeks, for the past month, over the past 48 hours regarding temperature and wind specifically when it comes to herbicide and herbicide efficacy. And then disease and insects are a little bit easier to rule out. Usually you can see evidence of what they're doing in the field. For those, I leave until the end. And then I start looking at inputs. Was the crop the appropriate choice for the field? Was there a residual herbicide product that was causing some issues from previous year? For our requirements met for fertility, did we put too much N down the seed row, for example? And was the variety appropriate for the situation at hand? I'm thinking clubroot tolerance specifically here. And then last thing, on to product performance. That's often the easiest one to jump to as a farmer. I applied this product, I paid 20 bucks an acre for it, it should work, you know, darn it. So why is it not working? It's rarely the product's fault in many cases. Usually it's a combination of other factors. Those products do tend to work quite well, and they have to be registered in order to be effective and prove efficacy. Pesticide products specifically. So then we start looking at things like drift, misapplied rates, mixing rates, incorrect orders of mixing, delayed activity, and then the crop stage timing. With those factors, usually we can come to a result or solution that gives us an indication what might be causing yield loss or less than ideal crop conditions. Now, this photo here was sent to me by Nicole, actually, last spring when there were issues with canola emergence in the Swan River Valley. And what really caught my attention here is how long the hypocotyl is. Now, let me just see if I can, here we go, show my red spotlight. I don't know if you can see my mouse on the screen, Nicole.
I can see your mouse, yeah.
Okay, yeah, I can't get the red little highlighter to show up, but anyway, you notice how long these hypocotyls are. It's a big, long primary stem before it comes out of the ground and turns green and chlorophylltic. Now, the seed at this point would have been based at the bottom of these hypocotyls. So you look how long they are. That gives me a dead giveaway that these seeds are too deep. They may not have been seeded that deeply initially, but due to a lot of soil and real estate changing hands in the valley the past year, there was a good chance a lot of those plants were buried, and they were buried deep as soil drifted over top of them. Soil drifts for a number of reasons: excessive tillage, lack of residue, windy conditions, those sorts of things. But keep that in mind when you're seeding into 2021. If those conditions are there again, perhaps seed a little shallower, seed a little later, and do a bit of a rain dance if that's what's necessary to get some water down. So these plants are certainly suffering from elongated hypocotyls, stress, and not enough energy to get to the surface. Some of these other ones are appearing to suffer from some sort of pinching off, some sort of root rot that could have been causing some effect there too. Some of these are growing and then turning upside down. Could have been a crusting soil surface, those sorts of things. So all things to examine when you're out in the field. Now, in terms of hidden yield loss and hidden yield robbers, disease is another one that tends to be easy to see, but hard to diagnose at the same time. Usually only see disease incidents when it's fairly severe and you're looking at the crop at harvest time and like, why is this crop so thin and so much more brittle and ripe than the other part of the crop? Those are easy to see. Early on in the season, it's a lot harder to tell. And I want to focus on a couple things here. We see the disease trends for the past 4 years in Manitoba through our disease survey. One of the big ones here is blackleg. We've had 3 or 4 relatively dry years in a row, with the exception of the fall of 2019 being quite wet for the most part. But in the growing season, it was quite dry, meaning that disease should be decreased. But in 2020, we saw the opposite, actually. We saw blackleg make a noticeable climb in terms of infection and incidence across all of Manitoba. But in the same breath, we saw sclerotinia decline. So we saw very little sclerotinia proportionally in 2020, but a lot more blackleg and a lot more verticillium wilt. This column is mislabeled. We're looking at verticillium stripe, pardon me, not wilt, stripe, on as a perhaps a significant yield robber, but it's a little bit too early to tell how it's impacting our yields, but it does make the crop tend to lodge and look ugly come harvest time. Now, if I start in the blackleg here, blackleg incidence in the Northwest region was at 84% in 2020 on 23% of infested canola crops with an average severity of 1. That would put blackleg severity right at this point, meaning 23% of every canola plant in an infested field where we found blackleg had a blackleg stem cross section of 1, meaning it's less than 25% of the cross section of the stem has Lepisvaria maculans causing yield loss issues. Now, blackleg is certainly one of those hidden yield robbers. For every step increase in blackleg severity, yield falls by 17%. It's not super noticeable in that first range, but as you get a little bit higher, 1.5, 2-ish, that drop becomes a lot more noticeable, and you certainly start to see that yield loss. Up here, when your canola is yielding 60 bushels and you might drop only down to 55, that's not particularly noticeable, given that you're still having a good canola yield. But as that starts to drop further and those blackleg incidence levels start to increase just due to tight rotations, resistant genetics not holding up, those sorts of things, blackleg is becoming more and more of a hidden yield robber. So if we take 17% off the yield off the top every single year by having average severity of about 1, how much more are we giving up by not having proper management? So that is certainly a hidden yield robber. And early on in the season, low level severity like the 1 rating here is not easily noticed. It's usually inside the stem. It may be on some of the lower leaves, but as the canopy grows, that plant is looking reasonably healthy. We only start to see those sorts of infections around swath timing and a little later when we start seeing discolored stems out in the field, or if you're actually doing your due diligence or your agronomist is doing that clipping of canola stems and actually examining the cross section. Usually we just look over the top, everything looks fine, and we move along. Doing a more diligent scouting job will really help determine how severe blackleg levels are, and then appropriate management can be taken earlier on. Blackleg is one of those diseases that needs to be taken care of and addressed early on in the season via genetics and fungicide application if necessary. Doing something late in the season is too late. That yield loss has already been set because these blackleg lesions are cutting off water and nutrient uptake and preventing grain fill in the plant. Now, I do want to focus on blackleg a little bit more. This question does come up quite often already, especially as different canola varieties have different ways of labeling blackleg resistance. So I wanted to address that and dispel a few myths about how resistance genes work in blackleg, because there's 2 different systems. Now, on the left-hand side of your screen, you're going to see what's called QTL, or a quantitative resistance, also called adult plant resistance or minor gene resistance. They all refer to the same thing. On the opposite side would be major gene resistance. Now, the one on the left is what you've been familiar with for quite a number of years already. If you're looking for a blackleg rating on a particular variety, it's going to give you a letter, R for resistant, MR for moderately resistant, MS for moderately susceptible, and S for susceptible. You're familiar with those R, MR, MS, S ratings. Now, these are rated as a percentage of the susceptible variety Westar. Westar is fairly old already, having not been grown commercially for quite a number of years, but is very, very susceptible to blackleg. And every variety is checked against that variety in terms of its resistance. So even if you get a variety that has an R rating for blackleg, it still could have up to 30% infection based on Westar. So anything less than, or 29.9, will allow it to get that R rating. So that doesn't mean it's immune and it should not have any at all, but there is always that chance. But this minor gene resistance strategy works as a whole plant kind of combination. It's expressed in an adult plant through the entire life cycle or life stage of the plant, but really this is where we're trying to see an overall level of infection. We can't attribute resistance to any one specific gene. So it's a combination of many genes. That's why it's called quantitative resistance. There's many different little hammers all pounding away on the blackleg infection to try and minimize it. And it doesn't localize it in a specific area. What it means is that it tries to slow and reduce the number of plant cells that die due to the blackleg infection that would otherwise be killed off in a susceptible hybrid. Using a different side of resistance, if we're going over to the major gene, that is expressed immediately upon infection in the plant. So if small 2 to 3 leaf canola plant is out in the field, gets bit by flea beetles, that allows an infection point for blackleg to move in off infected stubble. And that can start infection really early on. But if that infection gene matches the same gene that the plant has for resistance and they line up like a puzzle piece, then resistance is going to start taking effect immediately. But what it does is it's a hypersensitive reaction. It doesn't mean the plant is immune. What it means is that plant, those plant cells immediately surrounding the point of infection go into a hyperactive death. So they all pre-program to kill themselves immediately surrounding that point of blackleg infection, meaning that blackleg infection cannot keep surviving. It cannot spread past that because it needs a living cell to survive off of. And by containing it and putting a fence around that infection point, it prevents that infection from spreading further. However, this major gene resistance is a matching gene strategy. The resistance is specific to a single gene that must match the blackleg race. That's fairly complicated to understand, but I've sketched out a little picture here. So on the left-hand side, we've got our blackleg resistance genes. These are these triangle-shaped tips. One is a pathotype 1, one's pathotype 2. Now they have to match with the canola resistance genes. If we're using the most common canola resistance gene called RLM3, these puzzle pieces don't match. And if they don't match, you're not going to have resistance occurring. They have to match in order for that resistance to take effect. But if they do match and the population in the field is resistance gene 1 and the resistance gene 1 you have in the canola matches, then that major gene expression is going to start working and be specific against that specific race of blackleg. Now that's a headful to understand there. So how does major gene resistance labeling work? You're going to start seeing this on more and more canola bags if you haven't already. And I pulled the top part of this slide off the Canola Council's website. They've got a nice graphic there. So think about it in terms of herbicide resistance or in herbicide groups. You know you've got a group 1 and a group 2 all the way up to group 27, I believe. Different types of herbicide, right? And they work against specific genes. But now you know that wild oats could easily be resistant to group 1 grassy herbicide. So then you're going to have to start using a group 3 or a group 2 or something like that. It has a different number, but that will work against that product. Blackleg resistance major genes are labeled in very similar fashion, except they're using alphabetical letters. Alphabetical letters A, B, C, D, E1, E2, F, G, and X. And there's a 2-part resistant label that's going to be used on major gene resistant canola. So what we've got here is our first part from our previous slide, where we're using this minor gene resistance with this RMRMSS system, and then we add in the 2-part major gene resistance. So if the canola plant contains resistance to RLM3 type, it's classed as resistant group C. Or if it has 2 of them in it, it might be groups B and C as demonstrated here. But if they don't match up, here we have blackleg gene 2, canola gene 3, it's not matching up. But we have blackleg gene 1, canola gene 1, that equals resistance group A. So that's then that major gene starts working, but only against that resistant group. But if we have something different in the field, say the majority of our blackleg population in the field ends up being RLM or AVR RLM9, but we have this gene in the field, they're not going to match, and that resistant won't start working. And then we're back to relying on this qualitative minor gene sort of resistance. So it's not necessary that one is better than the other, but they do work in combination and in tandem. So that's an important thing to consider. Nicole did mention That there was a question on keepingitclean.ca and how that is related to blackleg resistance labeling. Keep It Clean is actually not related at all. It is about using registered seed products and pesticides and managing the spray to swath and spray to harvest interval. So it's not related to blackleg resistance labeling. So there's a difference there. Now, the reason I have this one circled is that most blackleg resistance in current canola varieties available on the market contains resistant group C, resistance group C. But the problem is it's not working as well as it should or well as it could, because only 5% of the blackleg population, more or less, depending on location in Western Canada, actually contains the corresponding AVR LM3 that would match resistance group C. And if they don't match, that major resistance group is less effective than it could be. And then that minor gene or adult plant resistance becomes more important as a genetic resistance strategy. So unless you know the major gene that's present in your field or the one that's dominant in the field, you have to make a match or it won't work. Now, how to choose a canola hybrid with that information? Different seed companies have different strategies. Some companies believe that they should label the major gene if it's present. Other companies believe that farmers don't know what genes or what blackleg races are present in their field, so it doesn't make a difference. They would rather then look at the whole field approach and stick with the RMR MSS scale using the minor gene approach. So the pros and cons to both, and it really depends what you're looking for as a farmer. If you are serious about your blackleg concerns and you know you've got a persistent blackleg issue that your traditional variety is not taking down, it's likely due to the fact that it contains resistance group C. Wanting to look at something different where it's labeled and then matching it with what you know is in your field by having that tested becomes important. Then you may want to look with one of the canola varieties that comes from the seed company on the left-hand side of the screen, where they label their hybrids. If you're not so concerned, or the blackleg resistance that you have has been working well and you use an integrated approach, perhaps it's fine to use a canola variety from the right-hand side of the screen. So it really depends on your management strategy and what your aims are. Using the major genes that are labeled becomes more effective once you know what's in the field. That is kind of a prerequisite. Otherwise, it's nice to have, but you don't really know how well it works. Okay, that's a bit of a harder hitting topic, but other hidden yield losses could be due to result from lodging and verticillium. So we did see a higher prevalence of verticillium stripe across Manitoba in 2020. It did seem to be exacerbated by heat and a lack of moisture in late July and into August, which made symptoms feel more severe. Now, not necessarily that they were contributing to yield losses, that we do see late infection of verticillium stripe for the most part, but the yield losses might have come just due to downed plants that tended to lodge and weren't able to be picked up as easily. And as a result, yield was left on the field in that case. So there were some questions, and there's a lot of research going into this right now. We don't have a lot of great answers. But David Kaminsky, I believe, is on the call, and he should address some of this a little bit later. Just a bit of a photo, which I believe he'll touch on too. Now, other hidden yield losses and things to be concerned about. Specific to northwestern Manitoba, you may have heard this already, but it's worth repeating, and I'm going to address it now with clubroot distribution. Where is clubroot found in Manitoba? In 2020, we added 2 new municipalities, arm of Roblin and the arm of Alexander. So in both northwest and northeastern Agro-Manitoba, where symptoms were observed. These were found in fields where galls were found on the plant roots. It wasn't just spores anymore. We are actually starting to see the galls on canola roots, meaning that yield loss is certainly starting to occur. So a big part of clubroot management is reducing the amount of a susceptible host, canola, being grown, and it means having a better rotation. So other oil seeds are not susceptible. Flax, for example, is a great alternative oil seed and has a good return on investment right now, especially with the way prices are going. This map is a little bit more harmonized with what Saskatchewan is doing, just showing where we know we have clubroot symptomatic fields and other fields where clubroot is present but hasn't shown up as galls on canola roots to date. So the northwestern part of Manitoba is certainly a higher risk, given that there's 2 municipalities there with known clubroot cases, and surrounding municipalities have a lot of canola being grown, as well as spores being detected. So that's important to consider and important for farmers to know that using resistant varieties sooner rather than later and taking proactive measures is going to become very, very important. So when you're looking for clubroot, where to scout? It's most often found by random happenstance by the farmer, just looking through the field, something was off in a spot. Because it tends to start off in patches and tends to start small, very easy to miss for the first couple of years. It's only when it becomes blown up in a bigger patch do we tend to start sitting up and taking note. So look for areas that are stressed, stunted, prematurely ripening. It can start infecting as early as a 6-leaf stage. It's most noticeable right around swath timing or slightly before. We have caught it as early as bloom stage, where we're regularly finding it. When it's smaller than 6-leaf or about 6-leaf, it's a little bit too small to tell unless you're looking very, very closely. So check field approaches, anywhere where equipment enters and exits the field, where soil can be brought in from outside fields on equipment. Field corners, low spots, drains, areas near yards and shelter belts, anywhere where soil and water tend to congregate and accumulate in a landscape. So that bathtub ring effect around a bit of a low patch, especially in a dry year. If that low patch can't be attributed to being drowned out, and it's still looking a little bit sad, a little bit sickly, as you can see in this top photo here, that's a dead giveaway that something's wrong. Go into that patch, make sure you clean your footwear after you come out, pulling up and digging those plants up actually so you can get a look at the roots and not rip the roots off is important. See what's happening there. If you start finding anything, give Nicole a call. She'll provide you with some resources as to how to deal with it, and probably put you in touch with me too. Now, the disease survey this year. We added 2 new RMs, as I mentioned, Roblin and Alexander. We had 3 fields reported to us outside the survey. We found 1 on our own. Good that it's still so low, but we want to stay proactive. We don't want to be picking up several hundred fields every year like they are in Alberta. We've had the luxury of learning from Alberta's mistakes, and they've had 20 years head start on us in terms of dealing with it. Let's not fall into the same path of complacency that might have happened further west in terms of kind of ignoring the problem until it gets on our own field. So here we've got this farmer in particular. It was an interesting story because they were seeding L233P, just trucking along. They didn't know they had a history of clubroot or know that clubroot was an issue. Happened to run out of seed, went back to his retailer. That's all they had left in the shed was 345 PC, a clubroot resistant variety. Kept right on seeding and happened to seed through the middle of a clubroot patch. So it's very clear cut as to what clubroot can do and what will do. On the left-hand side of the screen, that canola yielded 38, 39 bushel an acre. It was a little drier. It didn't do as well as it could have, but it didn't have any clubroot galls on the roots. Look on the right, plants didn't even survive until harvest with a susceptible variety there. Further back, they did, but the galls were severe and yield losses were significantly reduced. I don't even know if they made 19 bushel on the areas immediately surrounding that patch. So that's a testament to what resistance will do and how well it works. The point is not to overuse resistance. It still works in combination with proper crop rotations, cleaning your equipment, those sorts of things. So how to manage clubroot spores? You got to keep a lid on them, right? Keep them low. Don't let them blow up to the point where infection and yield loss can start to occur. So the first strategy is to use a proper crop rotation, a minimum 2-year break between canola crops. I know it's very hard to say to a room full of farmers when canola is worth 19 bucks a bushel, as I got texted to me today. I understand why farmers want to grow canola, and it makes a lot of sense. But in order to keep growing canola profitably, there needs to be good management. Just like with anything else, too much of a good thing becomes a bad thing. And having a decent break between canola crops, at least 2 years, it becomes very, very important. That allows spores to die off naturally, hopefully to the point where it's below the point of infection and yield loss. Scouting becomes crucial because clubroot tends to start in small patches. We got to look at every canola field every year. If you're not scouting, I strongly advise you to consider getting an agronomist in the field to take a good look at your canola crops. grow clubroot resistant hybrids. If you're in a high risk area, resistance should be deployed on all the acres if the risk is present. Because that same equipment is moving from field to field, it could easily be spreading around. Using a resistant variety is a great way to preventatively and proactively help reduce the risk. Control host weeds in your other crops. Good news is that list of weeds are reasonably easy to control with herbicide. So making sure that they are taken care of in those wheat, barley, oat, peas, and soybean years allows them to reduce that green bridge. Don't allow reinfection from year to year. And then if you have a patch, if it's detected early, using things like lime, grass cover, seeding it down to a perennial grass for a couple of years, helping reduce spore loads and reduce that soil from moving off site becomes important. This is what I want you to do. It's got to keep a lid. It's got to keep banging down that clubroot so it doesn't blow up in population to become a serious problem. It can exist in fields at low levels without seeing yield loss. So if we can keep it at those low levels and keep knocking it on the head, that'll help. The other half of that messaging is keep it low, is keep it local. Sanitation of equipment, trying to clean equipment off as best you can, knock off the dirt before you leave the field. That becomes absolutely critical to prevent soil introduction into new fields from infected fields. Patch management, if you know you've got a patch, it's really strongly advised to seed a perennial grass or something to put some cover on that spot to prevent soil from moving out and to prevent machinery from working through it. It doesn't spread if it can't be moved on soil. So that's the way to reduce it. And then reducing tillage. Obviously a good practice for a number of reasons, but if in terms of clubroot, the less you till, the less likely it's to spread. I often get asked about preemptively using clubroot resistant varieties. Is it a good idea if I don't have clubroot? Am I wearing down the resistance if I'm using it and I don't have clubroot? No, that's not the case, because that clubroot resistant gene only starts being activated if there is clubroot in the field. If there's no clubroot in the field, no selection pressure is activated, and there's no chance that those spores are able to overcome that resistance. So if the spore load is relatively low, that selection pressure is a lot lower too. So we want to deploy that preemptively in those regions where individual or regional risk factors are higher. I wouldn't classify the entire northwest of Manitoba as a high risk zone, given the amount of canola that's grown every year. So using clubroot resistant varieties becomes very important, as well as rotating resistant varieties. Making sure that that resistance continues to hold up is important. Where to find more information, the Manitoba Seed Guide, which was mailed out at end of November, beginning of December, has a page listing all the commercially available clubroot hybrids in Western Canada. There's quite a few there. That list gets longer every year, and there's many more Liberty Link and Clearfield tolerant hybrids now available. Used to only be Roundup Ready, but that list has grown significantly in the past couple of years. Now I talked about testing. Testing for blackleg, and you can also test for clubroot. If you're a member of the Manitoba Canola Growers Association, you pay your checkoff dollars to them. They are offering a free testing program for clubroot, blackleg, and blackleg race identification, glyphosate-resistant kochia, and verticillium stripe. So agronomists are welcome to test off on a grower's behalf as well. Visit their website at canolagrowers.com to take a look and see how that program can work for you. Here is an example report for clubroot coming out of the PSI lab. For this particular instance, the soil tested at only 3,328 spores per gram of soil. So a fairly low number, but that is a great way to detect it early, to know that yes, clubroot is present in this field, and 1 or 2 susceptible canola crops later means that this spore load at 3,000 could easily be blown up to 3 million spores per gram of soil. And once you cross this 80,000 spore per gram of soil threshold, thereabouts, that's when we start to see infection caused and galls forming and yield loss start to occur. So if we can keep a lid on it and keep it under 80,000, test early, detect it early, that'll really help reduce the risk for future years. In terms of blackleg, that same lab offers a test. Result comes back like this. It gives me an indication of what pathogen what pathotypes are estimated to be in the field. So the blackleg race phenotype was a phenotype 1, 4, 5, 6, 7, and 11. So blackleg was confirmed in this particular field. Oops. And the recommendation is to choose a resistant or moderately resistant variety with an R gene group. So that's your major gene group other than group C, because group C, that RLM3 was not working. So looking at something a little bit different, it would be more beneficial. So it's got to match these phenotypes 1, 4, 5, 6, 7, and 11. If I could find a canola variety that had a major gene that matched those, that would help reduce blackleg in the field in question. This test also determined that verticillium stripe was detected on almost 48% of the stems, 3 of the 8 ones that were submitted. So that was certainly present in the field and something we need to keep an eye on. Nicole asked me to address canola seed size. If seed size makes an issue or causes an issue for germination and emergence, specifically as it regards to Invigor canola varieties, they've gone with the Invigor rate system, where they have labeled bags A through D, D being the biggest canola seed size. Is a D bag going to be a higher germinating or a more vigorous canola than an A canola seed size? Well, based on a Ag Canada study done in 2015 by Neil Harker, they concluded that seed size or 1,000 seed weight did not have any significant impact on emergence, yield, or seed quality. This was based on seed sizes ranging from 3.96 grams to 5.7 grams, with germination exceeding 97% in every case of a single variety. However, the study did show that plants from the larger seed grouping did have increased early season biomass, and their flowering period ended a little bit sooner. So that can be interpreted as a larger seed has a little bit more nutrient reserves in that seed, just based on seed size. So that can help propel the plant to a bigger plant earlier on, but that can also be compensated by increasing your seeding rate a little with a smaller seed size. So you just get more biomass by having more, though smaller plants. It does not translate into yield at the end of the day, but that can make a difference in terms of flea beetle feeding, those sorts of things. If you have more biomass in the field, there's more options for the flea beetles to choose from, and less likelihood that they're going to kill any one individual plant. So that might make a difference. But in terms of yield, emergence, and quality, it does not. But I did talk about seed establishment a little. If you're looking
Can I interrupt for a second? Do you have a lot left?
No, I'm almost done.
Okay, just checking. Thanks.
In terms of seeding rate, then check out the calculators at canolacouncil.org/calculator. There's a nice seeding rate calculator. That's what's on your screen right now. If you have a number of different bags of canola seed, you can punch in your numbers here by a seeding rate, emergence, or density, and it'll spit out how many pounds per acre should be going down on the ground. We also have a reseed calculator from Manitoba Agriculture that if you do run into the unfortunate situation of having to reseed, it does work out a lot of the economics for you there. You can punch in a number of variables determining whether it's more advantageous to keep the crop or to pull the trigger and reseed. So that's all I had for right now. If you had questions, I'd love to take them, or send me an email.
Well, thank you, Dane, for the insightful information on canola yield robbers. And just a reminder for anybody that has questions, please put them into the chat box. Our next presenter is John Glowowski. John is our provincial entomologist and brings a wealth of knowledge to the Northwest workshop today. So with that, please welcome John.
Okay, thank you very much. And just a short bit of info about myself, since we were talking about shirts earlier. I don't have any Hawaiian themed shirts like Dennis, but I do have a rather large collection of both dress and t-shirts with insects on them. And normally I wear those on days when I do presentations. They're my good luck shirts, but being virtual, I did not wear one today. So if something goes wrong, it's because I'm not wearing one of my good luck insect shirts. So what we'll be discussing are 4 different insects, and the questions I got on these are for flea beetles, a little bit about the different species, some of the treatments that are available, especially some of the seed treatments, what's the difference between them. So we'll cover that. For cutworms, we'll look at things like why cutworms end up in particular fields, and again, treatment options. What can you do to control them? Are seed treatments an option? For grasshoppers, we're going to look at how to control them, the effects of weather, and how you can reduce your spraying load somewhat. And we'll look at, are wireworms an increasing threat in Manitoba? So to start with, flea beetles and different species. So there's actually 72 different species of flea beetles in Manitoba. And of those 72, 10 potentially would feed in a canola field. There's flea beetles that will feed in other crops and not touch canola. Some of them even look like the ones you see in canola. There's also beneficial flea beetles in Manitoba. In fact, there are species that were purposely released to control leafy spurge. So they might look similar to our crucifer flea beetle, but they would only be on leafy spurge. So just so I don't totally give flea beetles a bad name, there are good flea beetles and there are crop destroying flea beetles. Now, the 10 that will feed on canola, 2 of those are quite common. The crucifer flea beetle, historically our most common species in Manitoba. And as you get into the parkland area and into the northwest, the striped flea beetle becomes increasingly a bit more abundant, especially early in the season. One of the difference between these two, early in the season, it'll be mainly all striped flea beetle. They're an earlier emerging species. So any flea beetles that you see in April and early May, usually the majority are going to be striped flea beetle in canola. Later in May, usually about mid to late May, you start to see more of the crucifer flea beetles. And once we get into June, it's often crucifer that dominates. So that's the different types. Now, there's research going on right now looking at, do they feed any differently on the plants? They're both defoliators. They've got, they're small beetles, small mouth parts. They make little pits in the cotyledons, and they will feed on the stems as well, which can be a real problem. Now, one of the things that we know is different between them, they're not affected equally by some of the seed treatments. And it's not resistance. There's been no genetic change in the beetles. But again, we've got lots of different types of flea beetles, and they're not all going to respond exactly the same to a given seed treatment. And research in Alberta, this is by Jim Tansey, his graduate work at University of Alberta. He found that crucifer flea beetle was killed more by the neonicotinoid seed treatments, that's your Helix, your Prosper, than was the striped flea beetle. So now, that doesn't mean that the neonicotinoid products, your Helix, your Prosper, don't work on striped flea beetle. They do, they just don't work quite as well as they will on crucifer flea beetle. So that is one difference between the two species. Now, regarding seed treatments and insecticides in general for flea beetles, I've provided a list here of all the different products that are available. So under the seed treatments, anything I have in black here is what we call a neonicotinoid. These are a base seed treatment that you will have on pretty much any canola seed you buy. You will either have Helix, Prosper, Nipsit, or Sombrero. Those are your neonic. You can add on Lumiderm, Fortenza Advance, or Buteo Start. So these are the add-ons in light orange. The black is your base, your neonic. So with your neonic, your base, you're probably getting somewhere around 3 weeks of protection from the time you seed. And the big question is, what is the advantage to the add-ons? Do we get extra protection? I've seen mixed results on this. I've seen some research that does suggest you might get a little bit longer protection, but then I've seen other studies, such as Bob Elliott's work out of Saskatoon, that showed that some of these add-ons really weren't giving you that much longer flea beetle protection. Now with the Lumiderm and the Fortenza, they're a chemical called cyantraniliprole. They are very good on cutworms. So if your goal is to try to control cutworms as well, you will get cutworm control. If there's cutworms in the field, you would get control with Lumiderm or Fortenza. With the neonics, you won't get much in the way of cutworm control. So that would be potentially one reason to use something like a Lumiderm or Fortenza Advanced. Butyrostart is quite a new chemical, and I'm told that it gives you some enhanced flea beetle control, but it is new, and I haven't seen much in the way of independent studies on this, so I don't really have a lot I can provide regarding that. Now, as far as foliar sprays go, anything in blue on my list here is what we call a pyrethroid. They're all in the same general grouping. They're generally quite cheap, and they all tend to have the same basic structure to them. They're different products, slightly different chemicals, but they kill the insects in the same way, basically. So one of the questions I often get is, what is the best foliar spray for flea beetles? And it's almost an impossible question to answer, because I've had growers tell me, some will say that Desis worked for them, but Matador didn't. Others will tell me Desis didn't work for them, but something like Pounce did. Then I'll have people tell me that Pounce didn't work for them and Desis did. It really does depend on who you talk to. These are all pyrethroids. They've got a little bit of residual to them. One thing though with that group of chemistry is, don't be out there spraying a pyrethroid once you get into the high 20s. So if we're getting into late May or early June, it's a hot day, you might want to wait till it cools down before you apply a pyrethroid. They don't seem to work as well at killing insects if the temperatures get a bit too hot. And I've already mentioned to expect roughly 3 weeks from your base seed treatment. And again, you'll hear and see different things about how long something like Helix or Prosper controls flea beetles. I pulled this quote from Jan Canolo. She's an entomologist at North Dakota State University, and she's done a lot of different work and trials on the neonics and other products. So independent work, and her advice to me was you can expect about 3 weeks, maybe 4 if the weather conditions aren't conducive for flea beetles. But that's from the time you seed, not from the time you start to see plants. And the problem that we get into is when you seed, and it takes several weeks to get from the time you seed until the 3 to 4 leaf stage. That's when we get people doing foliar spraying for flea beetles. If by chance you can get to the 3 to 4 leaf stage within about 3 weeks of seeding, you probably don't need to be doing any foliar sprays. But when the plants get stuck in either the seed or seedling stage for too long, that's what's going to put you at risk of needing to do foliar sprays. And also with the neonicotinoids, weather does affect how well they work. This work was done by Bob Elliott at Ag Canada in Saskatoon, and he found that the neonic seed treatments were more effective in drier soils than in wet soils, and they were more effective at the warmer temperatures than they were at the cooler temperatures. So both soil moisture and temperature might affect slightly how well your base seed treatments work for you. Now, date of seeding and flea beetles. There's actually been several studies looking at seeding dates and your risk for flea beetle damage. But keep in mind what I said earlier, that what really matters is getting from seeding to the 3 to 4 leaf stage quickly. That's what really matters. Now, in the study that was done in Manitoba by Bob Lamb back in the 80s, they found that the earlier seeded plots suffered more flea beetle damage, likely because they sat longer in the field in the vulnerable stage. Jan Knodel, again in North Dakota, she did a very similar study a little while ago, and she found the same thing. Early seeding increased injury by flea beetles. Hector Carcamo, almost the exact same study at about the same time as Jan's in Alberta, and he got mixed results. It depended on where they were in Alberta. The earlier seeding reduced flea beetle injury in southern Alberta, but increased it in central and northern Alberta. So, but once again, the big thing is how quickly are those plants going to get to that 3 to 4 leaf stage, and will they be in that vulnerable stage at the peak of your flea beetle populations? Those are probably the two things that will determine whether or not you end up doing foliar spraying. So hopefully that covers the flea beetle questions that people had. So we'll move into cutworms. And now 2020 was still a bad year for cutworms. 2019 was probably the worst cutworm year that I've ever seen in my career. 2020, still a major cutworm problem, but maybe not quite as bad as 2019. So I'm hoping that our populations have somewhat peaked and are starting to decline, but that remains to be seen. So do be on the alert for cutworms when you're doing your crop scouting. And I always like to mention, just like flea beetles, when we talk cutworms, we're not talking about one species. In this case, we're talking about multiple species again. And the two that we've been finding recently at bigger levels were dingy cutworm, the one on the left in your screen, and redback cutworm. There are differences. Dingy cutworm is a climbing cutworm. It does very little clipping. So if you're scouting and you start seeing chewed up plants, but not plants lying on the ground, like you would expect for some cutworms, do dig around those plants. It could be dingy cutworm doing the damage. Redback cutworm, when they get bigger, they will do a lot of clipping, and then you know you've got a cutworm issue. So regarding controls, both foliar sprays and seed treatments, this is your list, and really there's only 8 active ingredients registered for cutworms and field crops in Canada. So list isn't great. What I have in blue here are your currently available seed treatment options. And these are all quite new. When Lumiderm came out in canola, it was actually the first of our seed treatments for cutworms, Lumiderm and Fortenza. And just very recently, within the last couple of years, Lumavia has come onto the market. And Lumavia is a seed treatment available in your cereals and pulses. The CPL stands for cereals, pulses, lentils, I believe. So that's what it's available in. And these products, Lumavia is a little bit different chemistry than the Lumiderm and Fortenza, but the same chemical grouping. They belong to a group called the diamides. Lumavia is the exact same chemical as Coragen, just as a seed treatment, and Coragen as a foliar spray. As far as foliar sprays go, things in this diamide group tend to have very good residual to them. So Coragen, very long residual. Same with Viego. It's a new product registered in corn and soybeans. It's probably being marketed more for the hort market than for field crops, but it does have a few field crop registrations. Coragen, a lot of field crop uses. Again, you'll pay a bit more for Coragen than you will for, say, one of the pyrethroids or for pyrophos, but you will get that long residual out of it. It's very good on the cutworm. So some trade-offs there in your decision making. The pyrethroids, same thing as I mentioned for cutworms. Just be careful when you're applying them. But for cutworms, you want to be spraying late in the day anyway. You don't want to be spraying midday or in the afternoon for cutworms. Cutworms are nocturnal. They spend the daytime hours hiding underneath debris in the field or in the soil. They come up at night and feed. So yeah, you don't want to be spraying for them in the day anyway. Make sure you're out there later in the day for best control. Although these products do have good residual, so if you did spray a bit earlier, they probably still would control things the next day. I have seen some head-to-head comparisons between some of these products. There's been some studies that have compared pyrethroids with organophosphates. In this case, it was Lorsban they were comparing with products like Desis and Pounce. And in general, the pyrethroids were more toxic and had a longer residual on the cutworms than the Lorsban did. So I know some people tend to pick favorites. In this case, again, the pyrethroids for cutworm control do seem to be a better option than the organophosphates. The diamides, I haven't seen them head-to-head with the pyrethroids, but they have a very long residual, so that would make an interesting comparison. The other point I'd like to make regarding cutworm control is not all cutworm species will be controlled equally by a given spray. You get species like the glassy cutworm. It's one that is a grass specialist, so you would find it in cereal fields, maybe corn. They don't come above the ground to feed much. They spend almost their entire larva life below the ground. It's really tricky to kill them. Red-backed and dingy cutworm that I showed you earlier, they do come above ground to feed, so you can kill them. You can get really good success killing dingy and red-backed cutworm, less so with glassy. And there's another species that's more common further west in the prairies called pale western cutworm. It really likes the dry areas of the prairies. It's also a species that doesn't come above ground much. So with pale western cutworm, it can be really frustrating when people are trying to control that with insecticides. Okay, so what determines where cutworms end up? So people were wondering about crop rotations. That can have a factor, but probably what really matters is what's happening in a field back in late summer, because cutworms, the pest species that we have, generally have one generation per year, and they're generally out laying their eggs in August, maybe into early September. So if you have a field that's dried down, matured, or harvested, not much there to draw a female cutworm in, the odds are you probably won't have as many in that field the next season. Now, crops that are staying in the field longer, whether it's say a hemp crop or a perennial crop, that's going to provide good coverage for cutworms, so that might entice them and draw them in. If there's a source of nectar for them, that might entice them into a field or a part of a field. I remember one time going out to a sunflower field that had just a patch of cutworms. There's, I think, about 10 acres or so that was really heavily infected. The rest of the field was okay. The farmer told me that thistle patch, where the cutworms were this year, was last year's thistle patch. And that made perfect sense. That thistle patch would have drawn in the females when they were laying their eggs. They would have been a good source of nectar for them. That's where they hung out and laid their eggs, and next season that was the cutworm patch. So cutworms can be patchy, and probably a lot does depend on what was present the previous season. Anecdotally, I had somebody who grew hemp on hemp in some plot trials and told me that where they had their hemp on hemp, that was by far their worst cutworm situation in all their plots that they had. There was something about the hemp on hemp that seemed to draw them in. I'm thinking it's because you had really good cover for them late in the season, and there might have been a food source for the adults as well. So again, just if you're trying to figure out why the cutworms are showing up in certain areas, those are some things you might want to consider. So we'll move into grasshoppers. And I always like to start by mentioning that we've actually got about 80 species of grasshoppers in Manitoba. They're not all crop pests. We have 4 species that we consider potential crop pests. Those 4 species will lay a lot of eggs if they get the opportunity, so they can build their populations quickly, and they will move into at least some, if not many, of your field crops. There's a lot of species that just can't or won't build their populations up or don't move into field crops. So bottom line, not all grasshoppers are a bad grasshopper, but you've got a few species that can do crop damage. In fact, there's even a species that feeds on nothing but Russian thistle. So technically that's a beneficial grasshopper. As far as the pest ones go, two-striped and clear-winged seem to be the 2 more dominant ones last year. Two-striped is a generalist, feeds on lots of different crops. Clear-winged is a grass specialist, It will feed on your cereal crops. It won't touch your canolas and soybeans and broadleaf plants. So if you do happen to know what species you got, that can help you decide what's more at risk. So the effects of weather on grasshoppers. We've had some rather dry summers recently. And if we get warm and dry spring and summer weather, you will get the grasshoppers emerging a bit earlier than they normally do and going through their stages a bit quicker. So they get into that adult stage quicker, and that might prolong the egg laying period. If you combine that with fall conditions that result in the first frost coming quite late, then you have a very prolonged egg laying period. And with some of our pest species, longer egg laying periods, they just continue laying more and more and more eggs. Some of the other species that we call non-pest species have a more finite number of eggs that they lay. The pest species, given the chance, can lay a lot of eggs. So that's one way that they're affected by the weather. Now, drought and grasshoppers.
John,
Again,
can I interrupt for a second?
Yeah, sure.
Are you almost wrapped up?
I have about 5 minutes left. Is that okay?
Okay, yep. Okay.
Or do you want me to move quicker?
Sure. It's up to you.
Okay.
Thanks.
Yeah, no, I will motor through this quick. One thing to consider if we get into a drought year, grasshoppers don't just feed on crops. In fact, our pest species do like a lot of the natural vegetation and will sometimes preferentially feed there. Problem is, you get a drought year and the grasshoppers start losing that natural vegetation. That forces them into your crops. And that's one of the reasons that we often see more grasshopper populations, or at least more damage to crops in those drought years. That natural vegetation is disappearing, and people are harvesting the natural vegetation from field edges, ditches, etc. Maybe I'll skip this one. I'm going to move along here. One thing you can do to try to reduce your risk: scout early. Look along field edges, ditches early in the season for tiny nymphs that are emerging. The top picture here is grasshopper eggs. That's how our pest species overwinter. When they hatch out, they're about the size of a wheat kernel. When they get to this stage here, where you can see little wing buds, not fully developed wings, just little buds, they're in their 3rd to 5th instars. That's the ideal stage to be controlling them if you've got really heavy populations, because by then, most of the egg hatch should be done. They can't fly yet, so if you have a population heavily concentrated around a field edge, they can't move far in when they don't have fully developed wings, and you might be able to do just the field edge, not a whole field. If you're having to treat a pasture or an area of a lower value crop, one suggestion that you could try is something called reduced area and agent treatments. This has been tested in Wyoming, where they sprayed strips, a pass treated, a pass untreated, etc., etc., etc. Intuitively, you might be thinking, well, you only get 50% control, but that's not the case. Grasshoppers move around a lot. They actually got about 80 to 95% control by just doing it in strips this way. So again, if it's a pasture situation, something don't want to spend a ton of money, that's a technique you can use. And if you apply this with a high residual product that hangs around a long time, you can reduce the costs by at least a half and still get some very good control. Another option is brand baits, such as Eco Brand. Municipalities have been using these ones in recent years for some of the roadside grasshopper control. One advantage is you can apply it to pastures while cattle are still grazing, and it won't harm bees. Problem is, you do have to apply it instead of spray it, so you need an applicator. The company that sells the bait will also sell applicators, or you can use a Valmar or something that puts on granular fertilizers that will also spread the bait. So different options. I should also mention some crops are less attractive to grasshoppers, oats being one of them, peas being another. Grasshoppers still will feed in these crops, they're just less attractive. So you might find that they're more in your wheat and some of your other crops earlier, but once those crops mature or are harvested, they're moving around looking for other things to feed on. That's when they move into things like your oats, your peas, possibly canola, soybeans. I'm going to skip the predator part. If you start seeing a lot of dead grasshoppers clinging to the top of your plants, there's a fungus called Entomophaga grilli. It kills the grasshoppers. They die at the top of the plants. You see more of this in a wet, damp year. So that's another reason grasshoppers do better in hot, dry years. There's less of this pathogen in their population. And the last question I had was wireworms. Are we seeing more of them? The short answer is no. There's been a big survey done in Western Canada. Our by far dominant species is one called Hypnoides bicolor, a less damaging species than some of the species further west in the prairies. I've been getting isolated reports of grasshopper damage over the past few years. There was a field of soybeans near in the eastern region that was reseeded last year, and I got a couple of reports from the southwest. But this was sort of normal, a normal year. I can't say that I've been seeing or hearing of any additional or more wireworm issues in Manitoba. Positive news, if you do think you've got some heavy wireworm populations, we now have a seed treatment and an in-furrow application that will control, not just control, but kill wireworms. The previous ones, like your Cruiser, only make them sick, they don't kill them. Broflanilide, a product that you'll see in cereals called Taraxa F4, actually kills wireworms. So if you're growing a cereal crop and you are seeding into land that you know has a high wireworm population, Taraxa could be an option. And in potatoes and corn, Samigra can be applied in furrow, and it will kill wireworms or corn rootworm. And I should mention that any of the insects I cover, we've got fact sheets on them on our website. So if you need more info, you can go there. And if you've got any questions and we have the time, I can take them.
Thank you, John. We always enjoy what you have to tell us about bugs, and sorry for rushing you through that last part. Up next, we have David Kaminsky, who is our provincial crop pathologist. And so let's see what he has to tell us about the crop diseases.
Okay, good afternoon, everybody. Hope you can see my first slide. And as I heard the other speakers talk, I decided to clip a few slides out of my talk. So I hope we can maybe get a little bit ahead on time and not go too long. This is just a quick disease update for some of those diseases that you haven't heard about or have only heard a little bit about. And we do surveying across the province, 3 major surveys, those being in canola, in cereals, particularly wheat, adding barley this year, and finally in soybeans. We did a little bit in corn as well last year. I'm going to jump to canola first of all. And Dane showed you this slide already, which was the prevalence of these diseases that we see year after year in canola. Prevalence is how the percentage of the fields that we looked at that had some level of disease. So it may not reflect the intensity. And Dane mentioned that blackleg may have had an uptick in the last year in 2020. And I want to contradict a little something that Dane said, and that was that the impact of moisture on blackleg, it is a residue-borne disease, and canola stem pieces hang around for a long time. So infection can occur in most years, and the impact is worse when it is dry throughout the season because it cuts off the water and nutrient conducting system, and that's why you'll see plants turning brown earlier in the year. You do see a similar thing with Sclerotinia stem rot, but it's not residue-borne, so it's somewhat influenced by the amount of sclerotia that are out there, the resting bodies, but also very significantly by the amount of moisture. And as you know, 2016 was a bit of a dry year. We still had the residual of the previous wet year, and the last 3 years have been quite dry. So the prevalence of Sclerotinia stem rot in the fields has been quite low. Verticillium, I corrected this. Dane said it was Fusarium, although he said there was some trouble with identification there. And yes, we made it difficult for our surveyors. Fusarium root rot is something we've been seeing year after year, but this year, for a change, most of this bar here was verticillium. Not so much identified by our surveyors, but we also send those cut stems to the PSI lab for blackleg identification of races, and they observed that verticillium stripe was in those stems as well, somewhere between 30 and 40%. Pod spot and aster yellows are not major things, so we won't talk about this. Why are we calling this disease verticillium stripe? A few years ago, you may have heard the term verticillium wilt. And first of all, let's talk about the earliest symptoms that you can see in the field. This is a one-sided discoloration of the stem. Unilateral discoloration is what we call it, but that's something that could be confused with sun scald. So if it's on every plant and it's in the direction of the southwest, be suspicious of something like the abiotic caused sun scald. Now, wilting has not been observed in Canada. It has been observed in Scandinavian countries where they grow primarily winter canola, and there the disease has a longer time to work on the plant and cause an actual wilt. This one-sided symptom is reminiscent of the fusarium wilt that, if you're old enough to remember, we saw back in the early 2000s, and it was restricted to one variety. This verticillium stripe does not seem to be fussy about which canola variety it's attacking. Now, Dane showed you a picture of a cross-section of a stem. Blackleg is much darker and generally a wedge out of the stem, whereas verticillium symptoms, these are the microsclerotia within the pith and the vascular tissue, they're much more diffuse. You can see a lot of these little black or gray dots just under the stem or within the stem cavity, and those are microsclerotia. They're survival structures, much like sclerotia in canola, but they're tiny by comparison. Here again is the unilateral discoloration. And here are a couple of symptoms that you might mistake for other diseases. The little microsclerotia that are just beneath the epidermis might look like the pycnidia of blackleg, especially the avirulent type of blackleg, which is Leptosphaeria biglobosa. And this shredding of the outside tissue, that's something that you might see from sclerotinia lesions, especially when the disease has started and then stopped because it's drier later in the season. But there's potential for this verticillium stripe to block the xylem or water conducting vessels, and we could eventually be seeing wilt symptoms in the field. For now, we're not, and that's why we're calling it verticillium stripe. Dane showed you this slide, and this is a stem that is opened up so you can see how prolific or plentiful those microsclerotia are, both beneath the epidermis, in the vascular tissue, and even in the pith cavity. And here's the comparison of verticillium on the left with blackleg in the middle. This stem probably has more than 50% of the vascular area that is damaged by blackleg, and it will act as a wilt later in the season. Again, what you'll see in the cross section with verticillium is a lot more diffuse, not these wedges or whole halves of the stem that are black. This is a stem that's healthy in cross section. Dennis talked a little bit about soybeans, and I'll just tell you that briefly the soybean disease results. And this in particular, the bacterial blight, is the disease we see most commonly year over year, and it thrives under wetter conditions, although it may disappear towards the end of the season as it gets drier. I want to point out that in the Northwest, although we only looked at 4 out of the 66 fields, we saw bacterial blight in every one of those and at a higher incidence than across the provincial average. Still, the severity, that is, the impact on the crop, is less than 2, so it's not something we worry about. These are the 3 major diseases that we see year over year in soybean. and they're at levels that I think fungicides are rarely justified. Here's another indication that it may have been wetter up in the Northwest. White mold on soybeans was found in half of the fields, but in those fields, only 2% of the plants had some level of infection. And we are seeing those less common diseases like frog eye leaf spot, even up in your territory. Let's switch now to cereal diseases, and I want to talk first about another bacterial disease, that is bacterial leaf streak, which was seen in wheat, and especially in one particular variety, that being AAC Viewfield. And where it was seen the worst was in an area that had some intense rainfall, as well as some hail during that time, and in some plots where fungicides had been applied a couple of times in the season, and yet we were seeing this kind of a burning of the leaves. Sent for diagnosis and found not to be any of the fungal diseases, which were controlled, but of course fungicides don't control bacterial diseases, so it still looked quite severe. And this disease can work its way up onto the heads and cause a discoloration up there, which is known as black chaff, that can also stain the seed. The diagnostic symptoms of bacterial leaf stripe, well, you have these early symptoms which look like water-soaked parts of the plant, and later on they begin to get a little darker. And as they darken, and when it's wet, you'll see bacteria oozing out of those lesioned areas and beating up on the surface of the leaf. When it gets dry again, those bacteria spread out and dry on the leaf surface. And if you hold the leaf at an oblique angle to the sun, you'll see kind of a reflective glint off of the leaf. We have a fact sheet on our website which tells about the sources of disease, and seed in this case turned out to be a prime determinant of where bacterial stripe was seen. The conditions that favor bacterial blights are very high relative humidity, which you'll find especially down within the canopy of the crop, wet weather, meaning intense showers and or hail, which damages the leaves and allows bacteria entry, and a cool spring, which we had in spades. As I mentioned before, warm, dry weather kind of stops the disease in its tracks, and you might see very little on the upper parts of the plant. But again, this is that plot of Viewfield that was sprayed a couple of times with fungicide, and there's a lot of bacterial stripe there. Just some pictures from our neighbors to the south in the University of Minnesota, where it is even wetter than it is most of the time in Manitoba. And these droplets on the leaf on the left are a combination of water droplets, which are clear, and bacterial droplets, which are kind of amber colored. And again, they spread out and dry on the leaf surface. Okay, we were asked about fungicides on oats. There are a lot of them, but people say they don't seem to make much of a difference. I'd like to point out that year over year, the most common disease we have seen in Manitoba on oats has been another bacterial blight. And of course, fungicides don't work on bacterial diseases. So that might be part of the issue. The other diseases that we see most commonly on oats are the Septoria leaf complex, crown rust, which we see a fair amount of on susceptible cultivars down here in the south, but I doubt that you see it very often in the northwest. And that's because the incidence of rust, crown rust on oats especially, is limited by the presence of alternate host. And that is a shrub called buckthorn, which grows along streams. And again, haven't seen much of that alternate host up in the north. This isn't a rust that blows into our region like leaf rust do. And finally, we have Fusarium, of course, on oats as well. Looking at what fungicides are registered on oats, there are actually quite a long list, up to about 25. However, a lot of those are only registered on one disease or another. And I'm looking at the ones that are registered on several, and their propensity for resistance development if they are used repeatedly. So first of all, these three are all group 11 fungicides. Those are strobilurins, and that's the class of fungicides that has the highest risk of resistance development. Vito is probably the one with the widest spectrum of control. It does include the Septoria complex, as well as rust if you had it, and a few other minor things, downy mildew. Propiconazole, which you'll know as Tilt, or many of its generic alternatives, is a group 3 fungicide, and they generally have a lower risk of resistance development if they're used repeatedly within the crop, and most of them control that Septoria complex. Then there are the products with a mixed mode of action, and we just have a few examples here, Delero, Quilt, and Trivapro. And with that mixed mode of action, they also have a lower risk of disease development. And finally, there is only one of the many fungicides registered on oats that is said to be able to suppress Fusarium head blight, and that is Carumba, which is a group 3 product. It also covers Septoria and crown rust. Now, multiple applications of fungicides on cereals, are they ever warranted economically? Well, in wheat, the main diseases that we deal with are the foliar diseases, tan spot and Septoria, and head blight. Your decision making when it comes to the leaf diseases is based on field observations, because these diseases start earlier and work their way up the canopy. They're not going to be that damaging down below, but if they got onto the flag leaf, that's where they would have the most impact. So you might use a fungicide, the T2 timing, that is flag half emerged, so that you coat the flag leaf and protect it. Fusarium head blight, on the other hand, we can't see it developing. We have to rely on risk forecasting. And the conditions that lead to infection are heat and humidity. Humidity above 85% and in the 15 to 30 degree range. The unique coincidence of those things is what leads to Fusarium infection. Now, we've been using the same model for many, many years, and I'd like to let you know that there is research into improving the risk forecasting. It's coordinated by Paul Bullock at the University of Manitoba, some of his associates there, and some of Manitoba Ag's people on the ground as well. Some recent results from the on-farm network trials, where on a large scale, growers have been applying fungicides aimed at controlling Fusarium head blight in particular, and they're looking at early timing, which is before many of the heads have emerged, to late timing, which is when the heads have emerged but have not yet flowered. And they found that there have been really no differences in Fusarium head blight. These are trials that have happened over the last 3 years, which I've emphasized have been very dry. There has been a slight yield increase, but it's not from the control of Fusarium head blight. It's from the control of those foliar diseases. Finally, I'd like to say that much research in the past has come to the conclusion that a fungicide at herbicide timing has never significantly boosted yield. It's the disease that gets up onto the flag, upper leaves, and the glooms that has some kind of impact. I have a feeling there might be a question or two that I have missed. So if somebody can audibly remind me of what those questions were, I'm just going to call up my sticky note that had them on there. And somebody help me out here. I'm stuck. Okay, so one question was whether it is necessary to use fungicides for Sclerotinia on canola in a very dry season. That was probably the #1 question that I was getting this year, as the spring was very dry throughout. And we've gotten pretty used to using Sclerotinia fungicides in a prophylactic manner, that is, whether the disease is there or not. But if it's very dry and the soil surface is staying dry all of the time, there really isn't much chance for sclerotia to germinate, for apothecia to emerge, and for ascospores to be blowing about. So in this last year, I think experience showed that when fungicides were not used, there was no impact on yield. But that's not going to be the case in every year. Each year is different, and whenever it's wet through the early parts, they have canola plants cabbaged out. By the time they're at 20% bloom and petals are beginning to fall, you're likely to have a high risk of disease. So I'll leave it at that. There's my contact information, and turn things over to our next speaker.
Thank you, David, for the update on crop diseases. Our next presenter is Kim Brown-Livingston. Kim is the provincial weeds specialist, and Kim has held a number of roles in the ag industry and is no stranger to Northwest producers in their fields. So with that, take it away, Kim.
Okay, thanks, Boris. I'm just going to get going here. I've got a timer set for myself because I know I had a lot of questions, and I did answer them all, and we don't have time to go through them all because I do have a lot of slides. So I'm going to go as far as I can till I run out of time, but I do have the information, and if anybody wants to Zoom me, we can go through the slides individually. If anybody wants to do that, we can do that quite nicely through Zoom or Teams or whatever is working for you guys. And like I said, if there's questions I can't get to, I certainly can go over them with you when we're off this call. So my screen is frozen. There we go. I was firstly going to talk about herbicide-resistant weeds, kochia, water hemp, and wild oats. And so I'll just go through a few slides on each of these. This is a graph from, there's a worldwide database of resistant weeds and to herbicides, and the number of species that are resistant to group 9, which is basically the class that glyphosate is in. And as you can see, there's quite a lot. This is when the first registered products came out in the mid-80s. Some of us will remember that. And we do see that shortly after that, within about just over 10 years or so, 10 to 15 years, we started to see the very first wild oat resistant species in here, which is this rigid ryegrass that showed up in Australia, and all through the years we've seen this. And in here, one thing to really notice, this is our water hemp. We first saw resistance to glyphosate in 2005. Our kochia is in here right here about between 2005, 2010, I think it was 2007, we found the first glyphosate resistant kochia. And as we keep going on up, there's lots of different a lot of grasses are resistant to glyphosate, and that does not bode well for wild oats. There has been resistance found to glyphosate in wild oats in Australia, and we don't have any of it here in Canada, but it was modeled that it was going to be the next one on the list, or it had a high, sorry, I guess I shouldn't say it like that, it had a high chance of becoming resistant to glyphosate at some point. So that is something that we really are going to have to watch, is our wild oats. So kochia with our Noxious Weeds Act, this is a tier 3 noxious weed. The Latin name is Kochia scoparia. It's an annual weed, but it can germinate all spring long or all summer long. Lots of seeds. The thing with this, it outcrosses, it either or self-pollinates, and that makes it a good candidate for herbicide resistance, because then we get a lot of different populations going, and the herbicide resistance can spread quicker. It can germinate again all through the summer, drought and salinity tolerant, and it can germinate very quickly in the spring. And as low as 21 plants per meter squared, so that's about 2 plants per square foot, can reduce wheat yield by 33%. There's lots of group 2 resistance. There's some group 4 resistance, not in Manitoba that we know about, but there's widespread group 9 resistance, which is our glyphosate resistance in Manitoba. So this is something to watch for when you're looking for glyphosate or for herbicide resistant weeds, when you see all stages of death. And so right here, we've got a little crispy critter, and right next to him, about a foot or 2 away in this soybean field, is a perfectly fine kochia, nothing wrong with him. This guy over here has been sprayed. He's pretty yellow. He's going to be absolutely fine. He's going to grow right through that. He looks a little bit sick, but he's going to be fine. but there's some carcasses right next to him. So again, you can see, and down here in this bottom picture, there's all kinds, you know, everything from this little bush plant here, and then this one is wilting and might die. I don't think so, but it's not looking great, but it may end up coming back. So we do see all stages of death, and that's a good indicator that there's herbicide resistance in there. The future of herbicide-resistant kochia in 2017, they looked at 305 sites in Alberta, and basically they sampled post-harvest in September and October, and they found that in 15 out of 16 municipalities there was glyphosate-resistant kochia. They found 100% were resistant to group 2, 50% were resistant to glyphosate, of the whole data set, and 8% were resistant to dicamba, which is group 4. That doesn't bode well for some of us that have started to use the Extend beans down in the southwest part of the province to combat some of the kochia issues down there, because there is group 9 resistant and group 2 resistant kochia, and an Extend bean uses dicamba as part of its herbicide platform. So they did find, and this is kind of scary, 10% of the samples that were tested out of the 305 were resistant to all 3. So we haven't hit that in Manitoba yet. I'll just show you the most recent testing that we did was in 2018. We did 300 sites in Manitoba, and then this is basically the map. And in 2016, the yellow is the bad stuff, the glyphosate resistant, and we see in 2016 there was a few spots in here, and by 2018, even a couple of years later, there's more. And this was a relatively small amount that was sampled. This just tells you where there was present or not. It doesn't tell you how many samples, but this is also only the samples that we did as Manitoba Agriculture. This doesn't include anything that farmers have seen or any testing that was done by independent agronomists or anybody in the industry. So this, moving on to water hemp, this is a map from Dr. Joe Eichley from North Dakota State University. This is all of the counties that water hemp occurs in North Dakota. And I'm not sure if you can see this, but it's pretty obvious that that comes right up to the border. So for us to think that it's not going to get here, I think is not a smart move. This is our water hemp distribution map in Manitoba. The map wasn't redone in 2020, just with a change in personnel at Manitoba Agriculture. And I will be updating the map. There's actually a couple more municipalities that have been found, as well as this circle up here in Dauphin. We found one plant of water hemp, confirmed water hemp plant in Dauphin, just a few miles north of town. We'll definitely be going back to that field and monitoring it. Hopefully, I think that field is going back into a cereal. We did find that water hemp plant in Clearfield canola, and it was surrounded by some waste high kochia as well. And so we're not sure where that plant came from. We just found the one, but I did get a call from a producer in the area telling me that he'd found a 7-foot tall weed and wasn't sure what to do with it. I was really glad he called me. I was working for crop insurance at the time as an insurance agent, but I was still really glad to be called and went out and took a look at it. And again, if you guys are seeing anything at all that looks suspicious, if you're seeing any pigweed patches that are looking funny or anything that gets sprayed, places to look for pigweed is, well, pigweed in general grows a lot of different places. Water hemp, because water's in the name, it does tend to like the wetter areas of the fields. You'll see it along the ditches, along the field edges, or along the drainage ditches. But having said that, it does still grow where it's quite dry. So that was us there in Carmen, there in that middle, that star. And again, we've got a couple more municipalities to add onto this map, and we're definitely finding more of it. This is kind of what it would look like growing very nicely in a soybean crop, and it is going to overpower that soybean crop very, very quickly. And this is after it was sprayed. This is after it was sprayed twice. So there's, yeah, it's a problem. This is, again, this is sprayed twice, and it looks like it just hasn't even stopped. So more than 20 plants per square foot can reduce soybean yields by 44%. That caused an average yield loss in corn of 18% on Ontario farms, all the way up to 84%. This is a really bad problem in Southern Ontario in their corn and soybean rotations. Over there, they tend to have a 3-year rotation with corn, soybeans, and winter wheat. And in the corn and soybean years, this is really becoming a problem. And it caused an average yield loss in soybean of 43% on Ontario farms, but that went all the way up to 93%. So early identification of this thing is the key. This is something off Twitter here that shows this on the left is red root pigweed. They tend to have more elongated cotyledons, which are the first seed leaves that come out, not the true leaves. And then they're more egg shaped, so they're a little rounder, I guess, for water hemp. And this is it. This is how small they can be when they first start out. With the pigweeds, I find you do tend to see this little bit of the red bit here in the notch. And yeah, but again, and if you pull them up, usually they have a bit of a red stem on them. So the problem is, though, there's a lot of different pigweeds, and when you're looking at them from here, they kind of all look similar. Red root pigweed, this is one that we're very familiar with. We've got smooth pigweed, not so much of a problem here. Powell, amaranth, not a problem here, and I hope it never becomes a problem. This is our water hemp down here, and you can see the leaves do tend to be a little bit longer. And our Palmer amaranth, and the biggest thing about Palmer amaranth is this petiole, which is the stem that the leaf attaches to the stem, or the stem of the leaf that attaches to the stem of the whole plant. It is longer than the leaf. So when you fold these leaves back, this little stem of the leaf itself is long. And so that's the easiest way to tell apart from water hemp and Palmer. And to tell both of those from a red root pigweed, basically water hemp and Palmer are very, very smooth and on almost, they can be a bit of a darker green color, but you can't always go by that, but they're almost shiny looking, because red root pigweed has a lot of little hairs all through it and water hemp does not. We do have a bit of Palmer amaranth in the province. It's just a plant here or there, but we're definitely watching for it as well. And this is a really good picture. This is what a water hemp stem looks like, very nice and smooth. This is a red root pigweed stem, very, very hairy. And the plant itself just looks a little bit duller because of the hairs that are even all over the leaves and stuff too. You can see a little bit here on these leaves. So one thing too, red root pigweed is monoecious, which means it has both male and female parts of the plant on the same plant. And water hemp and Palmer amaranth are dioecious. So there's male plants and female plants. And up here in the parkland, we see that with hemp sometimes. We see the male plants and the female plants, and sometimes they have both. But this is a plant that is exclusively male or female. And because of that, we get our resistance happening a lot faster, because we can have a susceptible female. And if she gets fertilized, if there's pollen from a resistant male in the area, that can fertilize that female plant, and then basic genetics will tell you that some of that offspring will be resistant, even though, whereas if that plant was self-pollinated, that whole female plant that was, that was setting seed, that would those would still be susceptible and we could still get rid of them. So that is why the resistance grows. It gets going really, really fast. There's some literature that says that using the same chemical grouping in the water hemp and the Palmer amaranth, using it continuously, we can develop resistance to herbicide grouping within 3 cycles of use on this plant. So it develops resistance very quickly. So we did find some of those spots in Manitoba. We tested 6 fields from the water hemp that was found in 2019 and did 5 samples within each of those fields. We found almost exclusively, other than a few samples over here, they were all resistant to group 9. That means glyphosate is not going to kill this. The group 2 resistance, there's a couple of different types of resistance they check for in the group 2s, because there's different types of group 2 chemistry that we spray. It's almost as bad. There's a couple here. This one, only 2 out of the 5 plants, but basically the rest of the water hemp plants were resistant to group 2s. So if we found water hemp in a field, I think automatically we could assume that it is going to be group 2 and group 9 resistance. It might not be, but I think it's probably a really smart move to assume it is. And again, the plant that we found in the field just north of Dauphin, that was in Clearfield canola. It had been sprayed, and obviously the kochia that was in that field, as well as the water hemp, were group 2 resistant. So what we need to do now is we need to look at herbicide layering using multiple active ingredients from different herbicide groups to control the same weed in the same field in the same year. That's not necessarily in the same tank. We're looking at using our pre-emerge herbicides, the ones with residual in them, and having that weed control extend through the crop into the growing crop, and then coming back in with our in-crop options. Now, this can also provide improved weed control and return on investment when there isn't resistance yet. And layering can be used to reduce multiple resistance in one weed or address several weeds, each prone to resistant in different herbicide groups. And we are seeing this down in the States. You are coming at this stuff with 4, 5, 6 chemical groupings. We're not spraying just 1 or 2 things anymore. You're seeing tank mixes with 2 to 4 or more products in that tank, and you're seeing a pre-herbicide with at least 2 to 3 products up front as well. And again, we just have to hit these things with a lot of different chemical groups to get rid of them. So what to do when you have water hemp, and I'm hoping it doesn't hit us yet, but we do know we've had 1 plant in the area, so we have to be watching. Initial testing results indicate a good chance of groups 2 and 9 resistance, so we need to assume it probably is. It does not appear to be a problem in competitive crops like wheat and canola. They just outcompete it and they get going. And what we can usually spray in those, especially in the wheat crops, our group 4s right now are doing a really good job of taking care of them. So we're doing all right in those crops. It's a serious problem in corn and soybeans, and we do have areas in the province that are heavy corn and soybean rotations. In fact, some of those rotations are just corn, soy, corn, soy, corn, soy. You basically have little to no in-crop options in some of those crops now. We do herbicide layering to get the weeds out earlier when those crops are non-competitive. Your in-crop options are limited, and in this case with soybeans, you're pretty much limited to a dicamba soybean if you're going to try to get that water hemp out of that crop. Moving on quickly to wild oats. This is the last herbicide resistant weed survey we did from 2016. We will be doing a province-wide survey again next year. It was slated for this year, but we weren't able to do it because of COVID last year. Normally Saskatchewan does theirs, followed by Manitoba the year later, followed by Alberta. And we've been doing that for a number of years. And so we've been doing weed surveys on a regular basis, but because of COVID, Saskatchewan's didn't get done. So everything's moved back 1 year. So you can expect to see us in the field looking for doing our regular weed survey and as well doing our resistant weed survey. So in the last weed survey that was done, which is 5 years ago now, we did find out of 79 fields, there was basically herbicide, there was group 1 resistant wild oats in 79 out of 101 fields in 51 municipalities out of 74 in Manitoba. Our group 2 was a little bit better, but we had 43 fields out of 101, which is about 43%. And that was in about half the municipalities. And the multiple resistance of group 1 and group 2 was 42 fields. So basically 42% of what we had surveyed was resistant to both groups 1 and group 2. And this is just a short list of some of the products. There's some other, these are just the main products, but there's also, you know, some generic and some trade names that are basically the same chemistries as these. If you are losing all of your group 1 and 2, if you have group 1 and 2 resistant wild oats in either wheat and or barley, basically all of these products are gone. You are left with Avidex and Fortress, which is a group 8 and a group 3 and an 8, and you'll get suppression with Focus and Fierce, and that is coming from this group 15 in here, this pyroxasulfone, and you're going to get suppression if you just use Treflan by itself. This group 15, we haven't been using it very long, and one of the problems is this is one of the few chemistries we have left to combat water hemp, and it's being used as a pre-herbicide ahead of our corn and soybeans as well in different formulations and in different chemistries, or sorry, different brand names. We're going to really have to watch using this group 15 because we don't want to overuse it. So multiple group resistant wild oats, basically there is groups that are resistant to 1, 2, 8, and 25, and there's also metabolic resistance where it just grows through the chemistry. So there's some wild oat populations out there that basically there's nothing left to kill them other than a group 15, and a group 15 on its own is suppression only, so you're still not going to get it all. There's a paper here. There was 1 field found in Manitoba. There is group 15 resistant to wild oats, but it wasn't, the group 15 chemistry wasn't sprayed in the field at that time. But when they were testing the wild oats in the lab and they were spraying it with different chemicals, it was resistant to group 15 there. So we didn't see it in the field, but it did show resistance when it was tested later on. So that is definitely something to watch out for, because like I said, if you have, that's one of the last groups left we have to go at, herbicide resistant wild oats. Some of the top 10 practices. maintaining a database so knowing what went where and knowing what's been going on. The data, on your own farm, plus there is an international database of just knowing what's going on worldwide with some of these resistant weeds. Strategic tillage, if, where, or when needed. Field and site-specific weed management, one size might not fit all, and I know lots of us like to have a wheat spray and a canola spray, and we kind of do all the acres of each crop with each one, with each what we've picked, and we may not be able to do that. And I think that's something that we really have to take a look at, and each individual fields have to have individual prescriptions. Weed sanitation, border control, that'll help slow down the herbicide-resistant weed dispersal. In crop wheat, use selective herbicides, rotate them. We're combating the non-target site resistance there. Herbicide group rotation, avoid back-to-back in crop group 1, group 2. And I know our farmers up in Swan River, when you're in wheat, you'll use a group 1 the 1 year, and then usually you go to a canola or a soybean, and the next time you're back into wheat, you go to a group 2 to get the wild oats. And that's, that's a really good idea to keep doing that. Herbicide mixtures and sequences are better than rotation, so throwing more things in the tank works better than just simply rotating away from a 1 into a 2 and back and forth. Pre and post herbicide scouting, know what's there before you spray and know what's there after you spray and what didn't work. Competitive crops and practices to promote competitiveness. Basically, it's kind of just a natural biological control. And crop diversity, obviously. Mother Nature loves diversity, and I'm sure lots of you have heard me say that over the years. So this is something that I'd like to just showcase. This is wild oat biomass at maturity, and this is either a continuous barley rotation, so this is coming out of Alberta, or a rotation where they did barley, canola, barley, peas. And they did a couple of different things. They did a seeding rate that was either low or high seeding rate. They did a short cultivar versus a tall cultivar. And then there was the combinations. So your worst case scenario on your continuous barley is low seeding rate. And even if you went to a high seeding rate, you reduced the amount of wild oats by 3 times. And if you went from a short cultivar to a tall cultivar, you reduced it by 2 times. So if you put both of those together, you reduced it by 8 times. And when you throw rotation in there as well, when you start rotating different crops, there's that diversity that Mother Nature loves. You actually reduce the wild oat biomass by 70 times if you're doing all of those things. So this is a tall crop at a high seeding rate in a good rotation. You've got 65 kilograms per hectare versus 4,500. So it's pretty dramatic. Putting all those factors together. And then this is the number of seeds, because the number of seeds that it produces will show, is adding to that weed seed bank and making our, 1 year, there's an old gardening saying that 1 year seeding is 7 years weeding, and it's exactly the same in crops. And so here, this is our continuous rotation versus our good rotation. And so when we went, if we went from a 1x to a 2x seeding rate, it was a 4 times reduction. A short to a tall cultivar was a 2 times reduction. And if you do all 3 factors, it's a 37 times reduction. So if you're looking here at a tall crop with a double at the high seeding rate, you have very few weeds per meter squared left, versus if you were in a continuous barley with a low seeding rate and a short crop, you've got over 3,000 seeds per meter squared. So that's a lot. Now I'll just keep going. And one thing I did want to mention, though, I guess just talking about this, I know we really like our short varieties of wheat. We like our Brandon, we like our Viewfield, and we are using our plant growth regulators to shorten them even further. And that is just something to keep in mind when we start to see these wild oat problems. A tall cultivar is much more competitive than the really short cultivars, and that's just something to keep in mind. But I know how well those semi-dwarf varieties do up here in the parkland. So I'll just keep going until Nicole tells me not to. These are some of the questions some more of the questions that I was asked to answer about spraying herbicides in general, everything from mixing order to tank clean out to temperatures, hot, cold, frost, to wind and inversions. So I'll start with mixing order. And I can send this out. This is actually from Jason DeVoe. He's with Sprayers 101. He and Tom Wolf have a web page. It's an awesome web page for agronomists and for farmers too. There's a lot of information there. This is the tank mix order. And if you remember this acronym, the W is wettable powders and flowables. Well, first of all, you'd throw some water in the tank. and then you put in your wettable powders, your flowables. After that, it's agitate, and at this point, it's your anti-flowing compounds in your buffers. If you're using those, there's a few herbicides that use those. And you agitate again after every time you add something into the tank. So if you're adding 2 or 3 products in, it's agitate every time. Your next one is your microcapsule suspension. There's not a lot of those, but that's something like your command. After that, you throw in your liquids, which is your liquids and your solubles. Your emulsifiable concentrates come next. That's most of our chemistry is an EC. And this is all in the guide. It tells us in the guide to crop protection what these are. And your high load glyphosates come in last. And the last thing to throw in is surfactants. There's a few times you should always consult the label. There's a few times like when you're with Amigo and Clethodim and Liberty, the order on that one is a little bit different, but most of the time your mixing order would be in this order. And every company as well has usually mixing order sheets or mixing order guides. Sometimes it's on an app. Corteva has a really good app. I haven't seen it updated for 2021, but I do have the 2020 app on my phone. It's really great. If you look at the mixing order, you pick your products and it tells you in the mixing order. So that's great. And if you look, BASF has it in their product book. Bayer has it in their product book. They all have places where we can find out what our mixing order is and when we're throwing in other things. If we are throwing in other things into the tank, make sure you get those in the right order, the odd time you're throwing in. If you're throwing in a fertilizer, usually it usually goes in last. But sometimes we're throwing in insecticides or some other things, and it's a good idea to consult the product label or talk to your rep about where we need to be at in the mixing order on those.
Hey Kim, do you have a lot to go, or are we okay to wrap up soon?
No, we can probably. I just basically I'll tell you the rest of the stuff I have. I'll flip through the slides just so you can see what I had. Sprayer clean out. There's a really good part on page 13, 14 of the guide to crop protection. There's 3 different kinds of rinses. There's ammonia, freshwater detergent. And when you look in the guide on every product, and here's an example with authority, there's the authority label, and second from the end, so right before the hazard ratings, it says sprayer cleaning, refer to method A on pages 13, 14, and then it'll say up on page 13, 14, it tells you method A, method B, method C. So most of the products in the guide have this sprayer cleaning, and if it doesn't have that, it will say refer to the manufacturer. So this is in the guide on every product page, but refer back to page 13, 14. And then after that, I just had some slides on, the spray drift and temperature and inversions and things, but we can go through that at another time. I did want to mention, though, our guide to crop protection is available now. The Dauphin office and the Swan River office do have them. They're $10, and I would get them as soon as you can. I would think we're going to have an early year, it looks like, and I would suggest getting a new guide every year, but they are available and they are at our ag office. And if you're not sure, you should phone ahead of time, but there is usually somebody in the office. And with COVID, we've had to be a little more careful how we have contact with you people, but we do have the guides, and they are for sale, and we'd like to get them in your hands as soon as possible. So you can contact either Nicole or myself about that if you're having trouble, or if you want some too, we can send them to you from Carmen as well. Just contact me about that. We can invoice you and get them sent out to some of the agronomists if you can get them that way. And that's all I have.
Yep, just to agree with you, Kim, just give me a shout. I have lots that I don't want to keep sitting on, so come and buy them. Thanks, Kim. It was nice to hear from you as our weed specialist, and your knowledge of a good number of fields in our area. And last up, we have John Hurd. John is our crop nutrition specialist for the province, and I'm sure John would way rather be driving up this way and get to test out his ice fishing, but he's going to have to plan for next year, I guess.
Yes, and for the second time in a row, Lori, I am not able to show my friggin' screen here.
Okay, go down to your PowerPoint again, John, your PowerPoint. Choose the other one. Choose that one. No, no, not that. Choose the other one. Yeah, try that one.
Okay. Does that one show up for you?
No, it doesn't.
No, it doesn't.
Can you go?
Well, just let me tell a story then, and I'll finish the wrap early, because thank God this is the last presentation I need to do this year, and I am just going to work through my pictures to myself, but I'll just tell people the story because I can keep it really short this way. So the first question was about phosphorus because it's expensive, and bottom line is, first good news is that the winter fertilizer ban is off. So if you are needing to get fertilizer out on your winter wheat or your rye, now's a good time to spread your nitrogen or forages. And it has been legal to go since March 23rd. And that's because there's very little snow or whatever, and we expect that you will be able, if there is rains, there'll be very little runoff, and nitrogen and phosphorus won't get into the surface water. So you are good to go for doing that or spreading manure. The other question that people had, what about the high price of fertilizers, particularly phosphorus? And I guess on that note, I have to say that not all farmers are dealing with high sticker shock right now. About over 80% of farmers typically purchase in the fall, and many apply in the fall. But for those that aren't, the last time we saw phosphorus prices this high was 2008. And we lived with that and we dealt with that. Phosphorus application rates were reduced a bit in spring 2009, but we toughed it out. What we did see is that application rates tended to stay low for a little bit after that. And that's because provincially people started growing soybeans and thought they could grow soybeans without phosphorus. Then we observed that the soil test phosphorus levels started to decline alarmingly. And since then, since 2012 or so, there's been a steady increase in the application of phosphorus. And I think the last few years, we have tried to remediate or replenish those depleted soils. And I think that tells us what our strategy is for this year, is that one option is to reduce your phosphorus rates, but in the long term, you know that you're going to have to replenish those rates in order to keep your soils productive. Like there's no other way to do that unless you're selling the farm. So my strategy suggestions I have, first of all, soil test, because that'll allow you to apportion which fields really need the phosphorus versus those that can get by with just a starter rate. And the soil test is the best way to do that. We've got research that shows that soils that have been built up to a medium-high range, they can produce some good yields with starter rates. 10, 20 pounds of phosphorus can still get good yields. But if our soils are testing less than 10 parts per million, I can't sugarcoat this. You need a full dose of phosphorus in order to achieve your yield potential. So those are the fields you're going to need your 35, 40 pounds of phosphorus on. That's the way that it is. And that's also the reward for those that have been able to take fields and over time build them into a 10 to 20 part per million range. There's some flexibility there in reducing rates. That flexibility really doesn't exist for us on our fixer-upper fields. Those are ones I would consider less than 10 parts per million. Up in your part of the world, 41% of the fields test below 10 parts per million, according to soil test surveys. And so those are the fields where you really need to target full rates. The impact of that, reducing it in canola and wheat can be dramatic, particularly if we're seeding early. Cold soils are notoriously low in phosphorus. We get low root growth and less diffusion. So early seeding should be coupled with phosphorus. If you're seeding, no, I shouldn't say this. If your neighbor is seeding in June, he probably doesn't need much phosphorus. Phosphorus is highly available in warm soils. And besides, the yield potential is reduced and you just don't need as much phosphorus when you're seeding late into warm soils. The next thing is, what about in the rotation? It's when we follow canola or summer fallow that we sometimes need to pay more attention to phosphorus. It's because if we're following canola, it takes a while for mycorrhizae to establish and recolonize roots and help with phosphorus uptake. So in those cases, if we're following canola, those might also be fields where phosphorus is impaired. And I know there aren't a lot of corn growers up there, but for corn growers, we've done studies, and those are places where you should not cut rates. Following canola, you need to keep your rates high in order to account for the lack of mycorrhizae. Similar would be for flax, another crop that's very dependent on mycorrhizae. We now have two approaches for phosphorus fertilization. We call what we've done historically is a short-term sufficiency approach. It's low rates of phosphorus, just enough for a good crop in the current year. It maximizes our returns, but does not consider the long-term value of the land or soil phosphorus levels. Under current systems, current yields, this approach tends to deplete the soils into a low range, but it's an appropriate practice in times of high phosphorus cost. What we're now doing in Manitoba, we're offering another option to farmers is we call the long-term sustainable approach, and that's where we actually manage to build phosphorus levels into a medium-high range and then maintain it there by fertilizing with according to removal amounts. Studies have tended to show that higher yield potentials are achieved if we do that, but in the short term, it's expensive. It's an investment in land, and when phosphorus prices are high, we say it may be better to use a short-term approach, and those are the sufficiency approach. Those are the rates that are currently printed in guidelines. Something else that depends on your opener. Those openers that tend to be at highest risk of seed burn or seed toxicity because of the tight placement of fertilizer with seed, for example, with disc drills, those are also the implements that are most efficient for phosphorus placement, that phosphorus uptake is maximized when we have that tight placement. We may not be able to apply full removal rates of phosphorus, but if you're applying lower rates of 10 to 20 pounds, those are the implements that make that phosphorus most effective. And the other thing I had is about the microbial products or enhanced efficiency fertilizers. A lot of this has been reviewed by Dr. Cindy Grant in recent publications, and Avail tends not to perform well in our environment, so should probably not be using much of that. Jumpstart, for example, or mycorrhizal inoculants tend to be very sporadic in any benefit. They may aid with phosphorus uptake, but they are not showing yield advantages in studies that have been done by Agriculture Canada and other institutions. So if I had scarce dollars, I would still be making sure I spend it on my phosphorus fertilizer. These products, all they do is they tap into phosphorus that you already have put in the soil, and if they help you use it up faster, you're just going to need to replenish it anyway. So I'm not really hip on using microbials to mine the soil further. Now, something you need to keep your eye on, and obviously many Manitoba growers have, when crop prices are high and fertilizer prices are modest, that's your economic signal that this is a good time to rebuild soils. And a really good time was last fall. It was the low phosphorus prices that triggered the US tariffs that caused the prices to go high. So whenever we do get that hint of good crop prices and modest fertilizer prices, that's kind of your signal that it's a rebuilding phase. Now, the other question that Boris wanted to stiff me with, because he already knows the answer, because the answer has not changed, and that is elemental sulfur is still elemental sulfur no matter how we dress it up. There is a process, or I guess that's it. The foolproof method to fertilize your crops like canola is with ammonium sulfate or ammonium thiosulfate, something already in the sulfate form. But sometimes those are inconvenient, especially if we're seed placing and it can cause seed injury. So if we don't have the imagination of putting our ammonium sulfate on in a fall band with MAP or nitrogen, or a spring band or a mid-row band or a side band, or even broadcast, because that's still a good way to put on sulfate. If those options are off the table and we're looking at other ways, there have been some great recent developments in formulations and some really good efforts to try to make elemental sulfur available faster. But they don't change the principles. So that the principles is that particles must be small in order for the bacteria to work on them. So first we need very small particles. And in order for particles to be small, we either start with big chunks and wait for them to degrade. The best place for them to degrade is at the soil surface. And that would be similar to a Tiger 90 that needs to that pellet needs to absorb moisture, a bit of frost action is good, and then split apart into those dusty type sizes. Those dusty type sizes are what's reactive and present. And a number of the new formulations have come up with those really small pieces, but we are not always leaving them dispersed through the soil. Sometimes they're banded. Sulfur bacteria must be present. We must have moisture. We must have oxygen. We must have heat because it's a biological transformation, and that takes place in warm soils much faster than in cool soils, and that takes time if we're in cool soils. And then we want to have that convert to sulfate in time for a hungry crop. Canola is hungry at the rosette stage. That's in mid-June. In corn, where we're finding we need more sulfur, the critical time then is V6. That's in July. I already got warm soils then. So elemental forms, I think, are probably, our friends in Minnesota are doing research, and they're finding that some of the new elemental forms are doing a good job at meeting corn requirements by mid-July. Not sure how we're doing yet with supplying canola by mid-June. So the new formulations, I don't think I could see, you're still not seeing my pictures, but there are several forms where they're applying as mixtures with other fertilizers. And yeah, be patient. These still need to convert to sulfate forms to be available for the crop. One of the new innovations that I think is going to be doing some research up in the Swan River Valley this year with Megan Rose is SolGrow 65, and it's actually a sprayable elemental sulfur that you basically run over your fields, I guess now or early, spray on these microscopically small sulfate particles, let them sit at the surface and work slowly, and then convert. And Alberta's studies showed that a good portion of that may still meet the needs of a canola crop. Other formulations are actually blends with urea or MAP, and those ones, it's a good thing they're small size, but they're still kind of buried in the soil with current practices, and their surface area exposure may not be as large as we would prefer, but again, they're in the soil, should be moist, but again, we're still waiting for warm soils for conversion. And that's about all I have to say about that. But I would field some questions if there's any. Oh, shoot. Hey, Kim, are you still online?
Are you going to talk about the CCA code,
CCA
John?
code. Up on, I got to get the CCA code.
I am.
Dan.
I am online if you need.
Lori said she can email it out with the recording as well.
Okay. Yeah. I sure am glad this is my last presentation of the year. I plan to do by webinar.
I'm glad you could wing it, John.
Well, I got to spend some time.
In the meantime, I could mention David Kaminsky just wanted to clarify something. He said he overlooked a question. It was, is very early seeded canola more prone to diseases? The short answer is no, except where soils are cold and emergence is slow. There might be more seedling rot and damping off. As long as you have enough plants to meet minimum plant stand, canola should compensate. Did I get that right, David?
Yes, that's right. Thanks, Nicole. Sorry, I'm a little slow on the switch.
That's okay. Thank you for clarifying. There was a question for John Gavlosky. I'm just going to find it here. You had described the practice of spraying for grasshoppers, only spraying every second pass, thus greatly reducing cost. Do you think there'd be a benefit of shutting off alternating boom sections on the sprayer versus leaving entire sprayer width passes unsprayed?
Yeah, I guess it can accomplish the same thing. I mean, ultimately what you're trying to do is reduce your insecticide load. It helps reduce the cost, but also it helps reduce the impacts of the insecticide on some of the beneficials. So that's what you're trying to accomplish while still achieving good grasshopper control. Now, in their studies, they did a few different trials, and they were often 100 feet swaths that they were using, but it doesn't really need to be that. I guess the big thing is you want the area to be large enough that you're leaving a little bit of refuge for some of the beneficials that might be present, things like some of the species of blister beetles that eat grasshopper eggs, beef flies, ground beetles, things like that, especially if you're using a very general insecticide. You want that refuge, and the grasshoppers will move around, so you'll get the kill. So it doesn't need to be 100 feet like in the diagram that I had. Smaller strips of refuge can work equally well and can achieve the same goal. So hopefully that answers the question.
Hey, thanks, John. And then the last question was for Dane, but Dane's not, he's offline right now. You mentioned the free testing offered to MCGA members. What would be most beneficial, to test current canola fields or to test those that are slated for canola the following year? And I think, David, you answered this one, or do you want to answer it?
Well, I did try to, Nicole. I'm not sure my response is complete enough. I said I'd suggest it's best to take your soil sample in the fall, soon after harvest, so you can get your results before you're making your variety decisions or even your planting decisions. But if you check your inbox, Nicole, Dane was able to send a more comprehensive answer for
yeah, I'm just reading it now.
And if there are others who are interested, especially in the resource materials he sent, perhaps they can get that from you.
Yeah, we can send that out for sure. If anybody's interested, they can contact me.
Okay, thanks.
So John, I see you got the code up.
Is that right? Well, we'll just call that dumb luck then. So.
All right, I think that wraps up the questions. I don't see any more that came in. So I guess, yeah, I guess that wraps up our afternoon. We did, we kept the afternoon pretty packed full of information, and we didn't really give you guys much of a break, but hopefully you enjoyed the afternoon. Thanks to Boris and to our provincial specialists for organizing this and tackling our questions. And for the presentations today, and we wish you all a great season and looking forward to working with you all. So I guess if anybody has any last minute questions, otherwise I think that's it. And the recording will be sent out to anybody registered.
Thanks, Nicole.
Thank you, guys.